Yeah, thank you, everybody. Welcome to US HUPO and the best of US HUPO today, 2022. This is our virtual day showing and featuring the best speakers that we had during our actual conference that we had in person in Charleston this year. But just in case that you have missed this or would just like to hear some of these talks again, thank you for joining the day together here with us. So we are trying to be more than just one conference in 1 week of the year, but really throughout the year offer you events, scientific symposia, webinars, and educational material. So I'm really excited about this super nice lineup of excellent speakers today. And we are also looking forward, of course, to next year being back in Chicago. But today, Ben Neely from Charleston will actually guide us through a day reviewing some of the fun events of our actual conference in March. So I'm turning this over to Ben. Thank you.
Thank you, Birgit, and thank you everyone for coming. We're going to get right down to it. So for the first speaker today, it's Susan Wild giving an encore of our keynote. Susan is from Georgia, and from the University of Georgia, and I'll let her introduce the talk and take it away.
Thank you, Ben. Can everyone hear me all right?
Perfect.
We can hear you.
Okay, great. Well, I was very happy to be invited to give a plenary at this meeting, even though I didn't really know what a HUPO was whenever Ben invited me. But now that I'm learning more, I realize that you all have very valuable tools and techniques to investigate further. We have answered some of the questions and as to why the eagles were dying, but now we have more questions, and I believe that your group will help us. So I'm going to back up a little bit and tell you what I'm talking about. This is avian vacuolar myelinopathy, or AVM, first occurred in Gray Lake, Arkansas, and we've conducted a lot of field studies and food chain transfer trials in the lab to find that we have expanding locations of this disease. We have completely characterized this new toxin and know now that other species are affected. I will talk you through some of these steps and then end with some of the human health concerns. This was published in Science, and we got to be on the cover because the eagle is cool. And they chose it as the best paper in Science last year and did this video describing it.
The AAAS Newcomb Cleveland Prize, our oldest award, recognizes an outstanding original research paper published in Science each year. The winning paper is chosen based on the quality of its scholarship, innovation, presentation, likelihood of influencing its field, and its wider interdisciplinary significance. This year, we present the prize to the paper entitled Hunting the Ego Killer. A cyanobacterial neurotoxin causes vacuolar myelinopathy, first published March 26, 2021. The paper was the product of an international collaboration co-led by Susan Wild, Associate Professor of Aquatic Science at the University of Georgia, and Timo Niedermeyer, Professor of Pharmacognosy at Martin Luther University, Halle-Wittenberg in Germany. At Gray Lake, Arkansas, in fall 1994, a mass die-off of bald eagles initiated a wildlife investigation. Dozens of birds died, and many displayed odd behavior, like missing their perches and flying into rock walls. When wildlife biologists examined dead eagles, they found extensive lesions throughout their brains and spinal cords. By 1998, scientists knew that birds from at least 10 different southeastern reservoirs in 5 states had this new disease called vacuolar myelinopathy, or VM, but no one knew the cause. In addition to avian species, Susan Wild and UGA colleagues documented that VM could affect amphibians, reptiles, and fish. Researchers made gradual progress uncovering the cause. Wild and her colleagues in South Carolina, where many of the birds suffered from VM, found dense growth of the invasive plant Hydrilla verticillata. The plant, imported from India for use in aquariums, was overrunning man-made lakes in the southeast, readily consumed by coots, which are frequently eaten by bald eagles. Using fluorescence microscopy, Wild observed that in affected water bodies, hydrilla leaves were densely covered with cyanobacteria colonies. This novel cyanobacterium was also found in all the reservoirs where the birds had died. Wild and her colleagues speculated that the cyanobacterium produced a toxin responsible for the eagle deaths. They fed plants collected from affected water bodies to laboratory birds, confirming the plants colonized by cyanobacteria caused VM. The cyanobacterium was named Autochthonous hydrolicola, Greek for eagle killer living on hydrilla. However, no known toxin was detected in the cyanobacterium. In Germany, Niedermeyer and co-authors grew this cyanobacterium in the lab. However, lab-cultured bacteria did not cause VM in test chickens. Hypothesizing that the toxin might be produced in lakes, but not in the lab, they collected more hydrilla leaves from impacted lakes. Direct analysis of the cyanobacteria growing on these leaves revealed a compound not present in lab-grown autochthonous that contained 5 bromine atoms. The team was able to reproduce the compound in the lab when the cyanobacteria were grown in the presence of bromide. The compound was revealed to be a novel biindole alkaloid, and further experiments confirmed it causes VM. The scientists called the compound autochtonotoxin, toxin that kills the eagle. Jana Marish at the Biology Center of the Czech Academy of Sciences led the genome sequencing of Autochthonous hydrolicola, which allowed the identification of the compound's biosynthetic gene cluster. Bromide can leach from rocks, but it can also be introduced into the environment from human activities such as coal-fueled power plants or water treatment facilities. The authors recommended increasing monitoring and public awareness campaigns be implemented for hydrilla and autochtonotoxin to protect both wildlife and human health. The study brought researchers together from multiple countries and disciplines to solve a decades-old mystery and reminds us how dangerous human-induced changes to ecosystems can be. These unanticipated effects are only likely to increase as we continue to alter the world around us. Hunting the eagle killer, a cyanobacterial neurotoxin, causes vacuolar myelinopathy. Recipient of this year's AAAS Newcomb Cleveland Prize.
There we go. So I hope I didn't seem braggy to play that video. I'm from the Midwest and we don't like to brag, but I thought they did a good job of giving you an overview of what has been done. And I will go back and show you a little more detail on some of the field and lab experiments, but I want to leave time at the end to tell you about some of the mammals that we have preliminary evidence that they're affected by this. So in 2014, we finally published the name of this new cyanobacteria that was growing on hydrilla in all the locations where eagles were dying. So we called it Autochthonous hydrilicola, eagle killer living on hydrilla. I love the video, but the pronunciations were a little off. So here's hydrilla. On the underside of the leaf, you can see spots with the naked eye. And under the microscope, if you mount them upside down, then you can see that that cyanobacteria is growing on the surface of the hydrilla leaf, but it's pretty cryptic since it's about the same color. So using fluorescence microscopy, we can sort of eliminate everything that doesn't have the phycocyanin pigments that will show up under this rhodamine filter set. So this is how we will screen the samples to find the cyanobacteria. It has a polysaccharide sheath. It does not come off the leaf very easily. So you really just have to look at intact leaves. And we've seen this disease expanding to new locations throughout the Southeast. And I must say that part of that is probably a matter of going out and actually testing and looking at these sites. But we have seen expansion of hydrilla, and I believe that the Autochthonous hydrilicola can also spread on other plants and through other means. We have noted that it is that many other taxa are vulnerable to this toxin through lab trials. This turtle was consuming hydrilla directly in a feeding trial for almost 3 months before it started to have this ataxia and weakness, and then anorexia. So this hind limb gait, having problems walking, it seems to be showing up in mammals as well. The tadpoles were killed by this toxin when they were consuming the hydrilla directly. They didn't survive the 30-day trial. And these little paedomorphic salamanders were consuming prey, the tadpoles and snails that were consuming hydrilla directly, and they were eating them. And they had seizures and paralysis and muscle atrophy, much like the birds do. And we noticed that in the amphibians, when the salamanders and in the snakes, that these lesions were presenting in the cervical spinal cord and in the olfactory nervous system. So probably their most important system for finding their food. And these were fed tilapia and fathead minnows that were feeding on hydrilla directly. And then once they're gut loaded with this prey, fed to snakes. This is a video of one of the sentinel mallards that was used out in a field trial, and he has had tremors and then seizures, and we euthanized him as soon as we took this video. We just wanted people to be able to see that the extent of this effect on them. And then in the end, he is almost paralyzed and his feet aren't doing any good anymore. So we were studying all these lakes across the Southeast. We knew they all had hydrilla. We all knew all of the places where birds were dying had Autochthonous hydrilla. And in one spot in particular, J. Strom-Thurmond Reservoir, is a very large reservoir, 70,000 acres, and they had extensive hydrilla. And from the time that it started growing densely in that reservoir, they were no longer having successful bald eagle nesting. So it was pretty obvious that they were a sentinel for this is poor ecological health. But they're attracted to these reservoirs. They like to eat coots, and sick coots are easier to catch than the normal silly coots. So I'm going to try to finish up this the toxin section so that I leave some time for questions, because I do want you to ask them. We start with a methanolic extract because you have to break open the cyanobacteria cells initially, and that works either for the plant autochthonous that you collect from the field or our cultures. And then we take it through solvent partitioning, and the lipid fraction goes on to the HPLC, and that fractionation went on to the mass spec to give us this molecular weight and a handy little formula that has a lot of bromide in it, 5 bromines. So in order to be able to determine which of the HPLC peaks was the toxic one, we needed a bioassay. So initially it was a cell culture, but that was identifying the presence of the cyanobacteria all the time. But there was something different that happened in the fall that it was more specific. If you used a Stereodaphnia, it wouldn't give you a false positive. It seems that it's always making this autochthonostatin, which is another compound or natural product that Autochthonous makes that we'll be talking about next year maybe. So here's this peak, and that peak is toxic to both the zebrafish and Stereodaphnia. So we knew that we needed to go after that peak with the NMR. The zebrafish, of course, are a pretty good model for human disease, and they have twitching, trouble turning right side up, some of the same things that we would see in the birds, basically, and they get tremors and very extensive seizures in the brain. There's a fairly low mortality rate in terms of how much extract needs to be in there to kill the zebrafish. 50 ppm is going to kill most of them, and that Bromide, the source of bromide in these reservoirs in the fall, when cyanobacteria starts to make the toxin, we'll say, well, where does that come from? Hydrilla can hyperaccumulate bromide and has quite high concentration of it. So when those plants are senescing in the fall, we believe that's the time when they would, the cyanobacteria can complete that toxin molecule. And we want to do some more work on that. The MALDI MS imaging that Timo Niedermeyer did over in Germany is fantastic, but I know we could do some additional work even with this toxin in tissues with folks in Charleston. So the first time that we did we were so ready we had the purified peak and we had enough of it to run a chicken trial, and it was negative. But once we figured out that in this culture media, there was no bromide provided. So once Stefan added Brenlinger added that potassium bromide, then we started to see this peak. And that culture now is producing a toxin, and that is the one that we did use on the second chicken trial. So this is what our molecule looks like, all those bromines. And we did get some clinical signs of neuropathy, as well as these lesions, which were clear in the cerebrum and cerebellar tracts. And if you take it up to the transmission electron microscopy level, you can see that these are intramyelinetic edemas. So this is a splitting open of the myelin. And we did find that toxin is in significant quantities in the fish that were collected from J. Strom Thurmond Reservoir, in the GI tract, as well as the muscle of game fish that are consumed by people. And in the wild coots, they had significant concentrations of a toxin in their tissues, in the breast and thigh meat, which again is consumed by people. So we want to be able to get rid of this toxin within an environment where it's present. And we're not going to get rid of all the hydrilla everywhere, but I think the only way to get rid of Beta-toxinus is to get rid of hydrilla in a situation where it grows very densely. And we're starting to understand which of our landscapes is really most likely to harbor this cyanobacteria if hydrilla grows there. And it's lower nutrients and lower acid systems, lower conductivity. It's a very interesting cyanobacteria. You can get rid of hydrilla using grass carp, and we have fed some grass carp to chickens, and the chickens didn't get lesions, but the fish do. They get really bad lesions if they're really small ones. So size is going to matter for this toxin, of course, just like many toxins. So now we have confirmed that this is a lipid-soluble toxin. This is not water-soluble. It doesn't break down easily. And we've confirmed trophic transfer and accumulation of this toxin in wild game and fish. So moving on, just want to show you a couple more recent information that we have using calcium imaging. You can see that these seizures last a long time. It's a really strong peak, and it just doesn't dim very quickly. But we have fully synthesized this toxin, and that means it's possible to do more experiments and learn more about it. So want to just move on and tell you maybe beavers get it too. We have evidence from the field that we have a lethargic clinical beaver. This is what beaver histology should look like. This is what the beaver that came out of J. Strom Thurmond looked like. So they definitely are showing up with vacuolar myelinopathy. And now we found the Florida panthers, who have been described as having this feline leukomyelopathy. It looks like VM2, and we found a toxin in their tissues. So this is ongoing work, and we'll see what happens there. But they have trouble walking. They have this hind limb weakness, and it's pretty classic presentation. Oh, don't want that. But these two kittens are still alive, and we could potentially get something from these kittens when they're going to put them to sleep for a little while, not kill them, but to take some samples and ask us if there's anything, because these kittens were obviously exposed, but they're starting to act more normal now. So I think I want to leave time for questions. So maybe I'll just end with this video, and then you can just watch the kittens playing. So this guy looks like he's pretty happy and fighting with something in his little cage, but his little panther kitten brother is kind of sacked out over there. All righty. I thank you for your attention. I welcome your questions. And let's see.
Thank you, Susan. I mean, it's a great talk and it's a great story. Something that we were kind of talking about is this idea of almost sublethal exposures. And I know it's hard to extrapolate, but like with the mammals,
Yes.
do you think, yeah, could you talk more about that?
Right. So Mark Cunningham has been working on this for a long time. And the first question, he's a vet with squidus, but he works down in, he's a vet with the University of Florida, sorry. And he also collaborates with the Southeastern Cooperative Wildlife Disease Study here in Athens. But he said, this is probably something they were exposed to. Like they have video of this animal FP256 from very early on. And so we weren't able to get the tissues and run those samples until 2 weeks ago, but they've been looking for anything else that could have possibly done it and really haven't found anything. So yeah, I think if they're consuming something in their environment in a lower level that they survive, but it's going to cause them to be debilitated and not be able to eat well, they're very skinny.
And not to be too focused. Oh, did I just freeze? Not to be too focused on the humans, but is there any spotty epidemiological evidence of people with mystery ailments? I mean, not to be alarmist, but that's not a stretch, right?
I don't think that it is. And I feel like the people who attended the conference have an even better idea of which of those neurologic diseases is there some possible like trigger that would be an environmental source of something that affected the system, and then they maybe have a genetic tendency, but aren't there several neurological diseases that we suspect are slightly different, idiopathic, and perhaps have an environmental trigger? So I'm trying to learn more about that, but I welcome anyone's ideas on something that might. I know a lot of fishermen that are having trouble with their eyesight and detached retinas. So I don't know, to me, it might be something in the optic region for people too.
And there was actually a question from the audience about that. But in your talk, one of the big kind of breakthroughs that enables these questions of like dosing or mechanisms is that you can synthesize it, right? I mean, that was kind of a big recent advance, right?
Yeah. Actually, that last paper was from Scripps. So it was a group of postdocs that were working on that, and they verified all of those gene clusters that would indeed make that molecule. So they did a really nice job. And we have the gene cluster for making a toxin now, and Linka is working on that in the Czech Republic, and we want to try to be able to use that as a tool, also a PCR technique to find AETX gene cluster in different places.
I was actually wondering, Peg, Susan, great talk. I was wondering, so you mentioned briefly, maybe there are still related other toxic compounds, right, that maybe are either metabolites of the existing ones that you identified. So are you, if you had to speculate, do you think there's much more than we think now, or and are you going after those?
Yes, and Timo is continuing to work on any other natural products discovery from them, and he's you know, this statin, his student Ricardo is working on that, and they should be able to get that publication together within the next month or so. But yeah, and in the panther tissues, there was an intact atoctinotoxin, and then the molecule minus a bromide, minus another bromide, minus another bromide. So when I was talking at the meeting, someone asked me, are you sure it's bromide that makes it toxic? Or is it that nitrile group or methyl group? Anyway, I have questions about that too.
Thank you.
And I guess one more, because I know there's a lot of ongoing work, but back to the mammals, because I think about mammals a lot. So, I mean, you're looking at the beaver, you're looking at the cats. I mean, have you gotten, I know you have to deal with a lot more approval, but have you had any samples of humans from these? I mean, that's, again, a big ask, but no.
I was working with some folks in California with other toxins, and so they were collecting stool samples and asked if I wanted them to send me, if they wanted to send stool samples to me. And I like collect wildlife scat all the time, but somehow that was more offensive. I don't know why, but it would probably be a way that you could perhaps look at it. I'm not sure, but it does seem like it's going to be in, it's in the eggs of a lot of the birds. So we think it might be that there's places that we could maybe milk, a mother's milk would be a way to look.
No, that's really, I keep thinking back to what the late 80s, the mystery Vancouver amnesia, shellfish amnesia poisoning, which was domoic acid, and you just didn't have samples. So it was all this circumstantially, I just wonder about these things happening and wherever across the Southeast, right? These are just in reservoirs. So.
Yeah. And I mean, it took me a really long time, as you've seen on the timeline, to get this far. And now that I think it might affect people, I want to hurry up and figure out if that's possible, at what level, and how we can avoid that. So it's my goal, and I would love for y'all to help me with this. And I want to go to Chicago. My daughter lives there.
Oh yes, please come to Chicago. Yes. No, I think it's really great to see how mass spectrometry can help in so many ways, right? And this really affects society and nature and the environment we're living in. And it becomes a much bigger scope than just research for academic aspects. So thank you, Susan, absolutely.
Thank you all for doing the hard initial science to get us to this point, so we can use these tools.
Well, yeah, please come to Charleston, but come to Chicago next year. You can come as a participant. And yeah, we'd love to have you again. So thank you, Susan. Appreciate it. It's great, as always. So I think we are ready for our next talk. This one is actually going to be recorded. So this is Chris Rose. I will let him introduce his talk, but he will be here for the Q&A after. So again, if you have some of these, I think it's something called single cell. I don't know, you've heard of it, but please post in the Q&A, and we will be getting to those, and Chris will be here to answer that. So I think we're ready.
Hello, everyone. Good morning from California. And thank you to the organizers for inviting me to speak here today to the Best of US HUPO 2022. I'm excited today to discuss some of the work that's been going on in my lab around intelligent data acquisition for multiplex single cell analyses. So at Genentech, we're very interested in working towards analyzing single cells. And a lot of this comes from the work that we've been doing at the bulk level for many years, where we work with collaborators to analyze the proteins and pathways that are going to be important in therapeutic research. And as we've performed this research, we found that collaborators often are looking to analyze smaller and smaller numbers of cells, where before we might have been able to analyze hundreds of thousands or millions of cells and get deep proteomic analysis, 8 to 10,000 proteins, that's quickly becoming smaller and smaller populations. And we're working towards the ability to look at single cells. And the case for this is really trying to understand cellular heterogeneity as well as rare cell populations and how those might be important in disease and or react to various therapeutics. So as we're doing this, our group is keenly interested in tackling some of the technical challenges associated with single cell proteomics, particularly some of the aspects of the limited proteomic depth, the fact that we can quantify about 1,000 to 1,500 proteins per cell, as well as some of the analysis of cellular signaling events like phosphorylation. Now, the main approach that most people in the community are using towards single cell proteomics is called SCOPE-MS. And this is a multiplex proteomics approach that uses isobaric labels and often adds what's called a carrier proteome into one of the isobaric labeled channels. Here, the carrier proteome in the first publication of SCOPE-MS contained 200 cells, and the rest of the channels contained 1 cell each. Now, the reason that we'd like to do this is because by adding the carrier proteome, you can direct the mass spectrometer where the peptides are in the spectrum, and then they can then be selected for MS-MS analysis and identification. And you can see here in the MS1 that the carrier proteome really makes up the majority of the signal. Really, without it, there's many peptides that wouldn't be detected. And then when we go to the MS-MS analysis, again, many of the fragment ions that are being used for identification are really coming from the carrier. The real challenge then becomes, how do we measure these small signals in the presence of this large one? So again, the advantages here are the multiplicative signal, the fact that we can identify peptides based on the fragment ion signal in the MS2, but of course, multiplexing requires additional sample preparation, and we have 2 challenges related to measuring smaller signals in the presence of larger signals. One is called ion coalescence, and this happens when two peaks are very close together, in this case 6 millidaltons for the N and C variants of the TMT tag. And I'll talk a little bit about this. And then also ion sampling, and that's purely how many of these single cell ions can we isolate from our sample while the data is being collected. So to touch a little bit on that, here I have an example of a carrier proteome that's spiked in either at 5, 25, 50, or 100x. And we have single cell proteomes as well, mixed 1-to-1. So in these cases, you can see here that we've kept the number of ions total constant, but we're increasing the number of carrier ions, which means we're decreasing the number of single cell ions. So as we increase the carrier proteome in this simulation, you can see that for a constant sampling amount, the variation of our measurement will go up because we're sampling fewer single cell ions. Now, this, of course, can be overcome by just sampling more ions. And in fact, if you sample something up to about 80% of the ions within this population, you can see that the variation is quite similar, although there's still a trend that when you have your higher carrier proteome, you're going to have more variation. We wanted to look at this in a more realistic sample. So we took a peptide in this case, and we labeled it with 50x carrier proteome in channel 126, and labeled the rest of the channels at a 1-to-1 ratio. We then analyzed this peptide over its elution profile with 3 different sampling values, and then looked at the quantification. And what was interesting is that we really saw this kind of stochastic detection of reporter ion channels from what we would deem to be single cell channels. And in this case, you can see that in one scan, only about 5 of them were present. In the next scan, many more of them were present. And if you looked at this, and you could mistakenly call that cellular heterogeneity, but at the end of the day, it really was just undersampling of the single cell ion population, because if you increase ion sampling, you can see that they're all there and that they have a much better coefficient of variation. Now, this relationship does relate somewhat to where it's selected in the peak, but there's a much better relationship of the variation in the measurement with the sum signal noise, which in our case is proportional to the number of ions that we measured. So we can clearly see that if we measure enough ions, our measurement gets much better, and we can start to use these principles to try to understand how the carrier proteome is impacting the quantitative values within our single cell analysis. So here we wanted to look at a more complex mixture now. So we took HeLa as well as K562, and we looked at carrier proteome levels from 5x all the way up to 2000x. And again, we saw the very same relationships, where if you increase the number of ions that you analyze, you decrease your measurement variability. We could also then look at if you called a threshold that you'd like to reach for quantification, in this case, 20% CV. We could then track how the number of ions you need to isolate changes as you increase the carrier proteome. And this is where we start to see that if you have something like 300x carrier proteome, you're going to need so many ions that it's going to be very difficult to obtain those ions in a single cell proteomics experiment. Now, while we were analyzing this, we also saw a quite interesting phenomenon in that when we looked only at the ions in the single cell channels, we found that once the ions got high enough, once we have collected enough ions in the single cell channels, we started to see increased variation. And this was quite opposite to what I'm showing you on the first plot, but the difference here is that this plot on the left-hand side includes both the single cell ions as well as the carrier ions. Here on the right, it only includes looking at the carrier ion quantification. So why are we seeing increased variation when we have more ions, which again is kind of counterintuitive? Well, to look at this more closely, we split up the channels, and we happen to have HeLa cells in the first 4 quantitative channels and K562 cells in the last 4 quantitative channels. So we split out the plots here for both HeLa and K562. And what I think is pretty clear is that when you look at this lower region, it's only the HeLa cells that are being affected. Now, we thought that this might be actually an interaction between this 127N channel and 127C channel. And when we remove that, you can see the effect is much reduced, but it's still somewhat present. And we think that this lasting increase in variation here with more ions is coming from interference from this 128C channel, because we're only seeing it with high levels of carrier proteome. But what this taught us was an important lesson that when you're using very high levels of carrier proteome, you really have to worry about the isotopes. The 127C is really the first isotope of the reporter ion that we used. In this case, it was 126. And the 128C is the second C13 isotope. So both of these happen to be causing challenges to the quantification in this experiment. Now, we took these learnings and we published a paper really around defining the carrier proteome limit for single cell proteomics. And it really was that. We really prescribed that for a single cell proteomics experiment, you don't go above 100x carrier proteome. And even when you're doing 100x carrier proteome, you're very careful about how you collect the data. Here we're showing that if you increase your AGC targets, basically you sample sufficiently and you apply post-analysis data thresholds in terms of the sum signal to noise that you're allowing through into your measurements, then you can drastically improve the quantitative performance or the quantitative rigor of your data. And of course, this will come at the cost of protein quantification events. However, in the case where you're really trying to understand which proteins are changing between 2 biological systems, this can be a really nice approach to confidently identify proteins that are differentiating between your 2 samples. Now, as we were thinking about how to better arrange an experiment around single cell proteomics, one of the main challenges that we kept coming back to was the fact that when you have a carrier proteome present, it allows you to sample and identify those peptides. But when you go to do quantification, you're still isolating the carrier proteome in the same isotopic cluster. And we thought perhaps we could take a cue from another technique that I have worked on in the past called Tomahawk, where we could use an offset carrier proteome that would allow us to do an MS2 on the carrier proteome alone. And then once we identify that peptide, we can do an offset MS2 and then potentially even MS3 on the endogenous peptides. The benefit here is that we wouldn't be co-isolating the carrier proteome when we're doing the quantification events. We could actually do that in the absence of the carrier proteome, but we would still be able to use the identifications from the carrier proteome when we're trying to understand which peptides to attribute that quantitation to. Now, the real challenge with this is that there wasn't a mechanism to do this online on the instrument. So to do that, we actually had to create an infrastructure to allow that type of experiment. And that infrastructure really came from taking a lot of the same principles that we have after we analyze data. In this case, we would have raw data, it gets sent off to the instrument computer. After that, it gets sent off to potentially some clusters where you do the search, and then eventually you interact with it as at your own computer. Now, there's a different paradigm that has been evolving for quite some time, where we're going to be analyzing the data in real time. And this is the approach that we've taken now to enable these types of experiments. So Thermo has an instrument API application where you can take raw data from the instrument as it's being collected. You can analyze it, such as getting a peptide identification to an actual spectrum. And then you can send back instructions on what you want the mass spectrometer to do. We've been using this ourselves for different applications, some in chemoproteomics, some for PTMs, and it's been attractive because it's allowed us to make our own custom acquisition methods. And one of the main applications where this has been really useful has been in the SPS-MS3 space. So in this case, we have, for SPS-MS3, it really works well because we have a survey scan and then an identification scan, which can be very fast, and we have quantification scans, which happen to be quite slow. Now, in a standard method, if you do this type of analysis, you have MS2s, and only about, say, 30% of those or 40% of those would actually be identified. And then after that, you would be taking MS3s. Now, in a normal method, the MS2s and MS3s that are identified are really wasted time on the instrument, particularly the MS3s, where the orbit trap time is very valuable because, again, these quantitative scans, which happen in the orbit trap, are quite slow. So in a real-time search paradigm, you can take all the MS2s you want in the ion trap. It's very fast. And if you identify a peptide, you then trigger the MS3. And it's only in cases where you've identified a peptide online that you will take the MS3. And that helps to improve the duty cycle and maximize the orbit trap time you have when you're actually trying to quantify peptides. So this has shown great strides and great improvements for SPS-MS3 analysis. And in fact, the API programs that really have sprung up have been from mostly Steve Geege's group and been dedicated to SPS-MS3 type applications, which include both just doing real-time database search plus MS3 through RTS-MS3, which was kind of the first implementation, Orbiter from Devin Schweppes' lab, which is kind of in a more advanced implementation, and then also Tomato, which is an API version of Tomahawk. So we wanted to create our own API program that would allow us then to do these more complex types of experiments. And we created a program called InSeq API. And InSeq API is a Windows application written in C#. It has a backend library called Scarif, which is a class library that helps us to create methods, and I'll get more into that a little bit later. And then it also enables us to output various files, which in our case is kind of an attractive aspect of this because we can output all of the results of every filter that was applied during the analysis. And we can also create methods which can be taken between instruments to allow us to do the similar type of analysis on any sort of instrument that we'd like. The real-time search is enabled with Comet. That's very much like Orbiter and in the commercial version of the real-time search. And the computer we used was the exact computer that came with the instrument with some upgraded RAM to the maximum amount that we could get it. So this is really the kind of special bit, I believe, of InSeq API, and that's this method structure. So this method structure is very similar to the methods that we have on the instrument, where we have nodes, we have filters, and then we have a scan. And the scans themselves, the raw data will come into a method, and it will go through this filter list, and then if it passes all the filters, we'll send back a scan. And it's flexible in that there is a filter store, which has various types of filters that we might want to use on the data. And we can arrange those how we'd like within a method and create custom methods in a very easy fashion. And this is just an example of what this looks like for a regular RTS SPS-MS3 method that would really be recapitulating what you can already do on the instrument. You can see here that we have an instrument ion trap MS2. We would have that spectra go through these various filters, and if it passes them all, we would send back an SPS-MS3 scan. So one of the main things we wanted to do was make sure that this processing was fast enough to be done in real time while we're collecting the data. So here is shown is the total processing time for a sample run. You can see here that the median time is about 5 milliseconds, which matches up well with the ion trap scans, which themselves are about 11 milliseconds in total. If we look at how that breaks down for all the various different filters that are being applied, you can see that the real-time search filter is taking up the majority of that time. We have slightly more time being taken up also by the LDA filter and some by this SPS ion filter, where we're going through the spectra and trying to match fragment ions. Now, one of the other important things that we'd like to look into is basically how this relates to the real-time thermal search, the type you could run on the instrument through the commercial version of the software. So here we have two runs, which aren't exactly aligned. We have a thermo RTS where we're looking at human canonical plus isoforms as well as decoys, about 40,000 entries. And for our purposes, a more complex database that included canonical plus isoforms and tremble as well as yeast and decoys. So it's about 4 times as many entries. And as you can see here, the search time on these is actually still quite short. We still have most of our search times under 10 milliseconds, even though within CKPI, the database is much more complex. Now, the one area where things seem to differ a little bit is the time between the identification and the MS3 scan. You can see here with the stock version of Thermo RTS, it usually happens under half a second, whereas with in CKPI, it's generally happening under a second from identification to MS3, and ours aligns much more with a regular SPS MS3 that doesn't have the real-time search component. So we're still looking into this and trying to see how we can optimize that. Now, lastly, I want to start getting into some of the comparisons between ScopeMS as well as this offset mass targeting approach, this tomahawk-like approach. So to do that, we have a ScopeMS sample, which is an 8-plex sample, and we have this offset mass targeting triggering sample. Sometimes you'll see this referred to as TAMA or tomahawk, and this was a 9-plex sample. These were all analyzed at 400 picograms for each of these, what we call single cell samples that are simulating single cells. And then the carrier levels were from 5 to 1,000x. And we analyzed these with 4 different methods. We analyzed it with a real-time search, where we do an ion trap scan and trigger an FTMS2 scan. That's also called a reticle scan by Erwin Schuf. We also looked at real-time search plus SPS-MS3 on the instrument. And then we looked at triggered offset MS2s as well as triggered offset SPS MS3s. And actually all of these were collected through NCKPI to keep things standardized, but you can see the parameters down here too. So again, you can see for the triggered offset, what we'd have is the first scan that comes through in the ion trap MS2, we take that through our filters and then we send an offset MS2 scan. And if we want to do an MS3, then we can actually send the very first MS2. This is the carrier proteome MS2. That goes through this first node in our analysis. We then send the offset ion trap MS2 scan. That gets taken. And then after that, that scan goes through this other node within our method. So this is a method now that has 2 nodes, one of which is looking for the carrier proteome. The second one is analyzing the offset ion trap MS2 scan. And once we get that, then that can give us back the offset MS3 scan. So when we look at the data, what we can find is that in general, the tomahawk approach, or this offset mass triggering approach, gives us more signal within our quantitative scans. And that's to be expected because we're no longer also isolating the carrier proteome within our quantitative scans. We are solely quantifying endogenous or single cell peptides. So the fact that we're getting more quantitative signal actually is not too much of a surprise. But you can see that's the case for both FTMS2 as well as SPS MS3 in this case. The tomahawk approach is much higher than the ScopeMS approach. When we look at how many proteins we quantified, it's actually quite interesting. You can see that at lower carrier proteome levels, ScopeMS would actually outperform this offset triggering approach. However, as you increase the carrier proteome level, you can see that we actually reach a plateau where there gets to be some sort of trade-off between how many scans we can acquire as well as how much signal we're getting from the single cell channels. But overall, you can see that we're getting more or less equivalent performance to scope, but we're less affected by the increase in carrier proteome level. And this is more or less recapitulated for the SPS MS3 data. Now, one of the things that we looked at in the Nature Methods paper was what if we change ion sampling parameters? How does that affect the quantification? Here, we're doing the same thing. So we have low ion sampling parameters, medium or high ion sampling parameters. In this case, you can see the trend holds for all of them, where somewhere around 100x carrier proteome, we have this kind of crossing of the two lines in terms of tomahawk or ScopeMS, and tomahawk is just much more resilient or agnostic of the carrier proteome level in terms of the maximum number of proteins that you can quantify at any given point. And generally as well, you'll see that if you look at the coefficient of variation for this 1-to-1 mixed sample, you can see that generally it's lower with tomahawk as well as compared to ScopeMS, showing that the quantitation should be better as well. To really get into the accuracy, we then looked at a 2-to-1 sample. So in this case, we have 2x sample in the last 4 channels and then 1x sample in the first 4. Here, the 1x sample is 400 picograms, where the 2x channels is 800 picograms. We have the same various carrier proteome levels here as well. And while the IDs showed somewhat similar trends, you can see here that we actually get a little bit of a variation, mostly due to the fact that we have more signal within those quantitative channels that actually is more beneficial to SCOPE-MS. But at the very high carrier proteome levels, you can see that we still see this increase with the tomahawk approach. But I think what I'd really like to demonstrate here is the fact that, as you see, even in the absence of interference, as we increase the carrier proteome amount, we're actually seeing this ratio compression within SCOPE-MS, and it starts to happen somewhere around 100x carrier proteome for both MS2 and MS3. And this really is interesting because we don't see the same thing with the offset carrier proteome approach. We actually see it increasing for MS2, which is likely contributions from the super heavy ion, which we're using for that offset mass triggering. If you use SPS MS3, this is actually reduced quite a bit. So what we think is happening then for this SCOPE-MS is really this idea that the higher carrier proteome levels are truncating our single cell signal. And this is also something that we saw in our Nature Methods paper, where if you look at ions in the single cell channels, you can see that at a high carrier level like 300x, it's actually muted as you look at the total number of ions. But if you exclude the carrier proteome from analysis by taking the ions into the ion trap, getting rid of the reporter ion for the carrier proteome, and then analyzing those ions in the orbit trap, you don't see that truncation. You actually see that it's a little bit more linear. And it really could be this kind of space charging type effect that's limiting and compressing the ratio here within our single cell quantitative channels. So this is kind of a new phenomenon that we think we've stumbled upon, and something also to potentially give us pause for going up too high in terms of the carrier proteome level for single cell analysis. Very lastly, what I'll say is that we're also taking this into real single cells. So everything I've shown you to date has been on lysates. In this case, we actually took K562 lysate, we labeled it with TMT Pro SH, and then used that within the Cellin-1 system and the proteochip as part of our master mix and digest solution. And then we did an offset carrier proteome analysis on real single cells. And again, we analyzed this with the INSEQ API triggered offset analysis. And in this case, we're still working on optimizing this method. We did see that in the single cell channels, we had enough quantification per channel to make us think that the cells were there. The reproducibility wasn't so hot, so we're still working on those approaches. But in general, you can see that the number of proteins is also somewhat similar to what we were seeing before, anywhere between potentially 750 and maybe 850 proteins once we apply our signal to noise filters. So this is something that we're currently working on optimizing. Once we finish up this experiment, then we'll wrap up this whole story. But we are excited about using this method going forward. So with that, I hope I've shown you that INSEQ API is really a flexible API interface that allows you to make many types of methods, and that this offset mass triggering approach is really a nice approach to single cell proteomics, particularly because it will help to decrease variability because more of the signal and more of the ion current we're selecting is on the endogenous peptides, but also that it helps maintain quantitative accuracy, most likely by not causing any sort of space charging effects when we're looking at the reporter ions, which in a SCOPE-MS experiment would include the carrier ions, which in our experiment don't. So with that, I'd like to thank the members of the group who helped out with this, particularly Tommy Chung, as well as those at Cellin-1 who have helped out with getting us going on the Cellin-1 system, and then all of the members of MPL NGS. And I would like to thank you all for your time.
Yeah, this was really great, Chris, and I see you're here live now with us. This is super exciting. And, you know, I had actually a question. Yeah, so it's fascinating what you show us. And of course, we'll all be reading the Nature Methods paper. But I wanted to ask you, so how do you anticipate it? How easy will it be for a lab to implement your workflow? I think one would have to also update the computer, as you said, for the computing power. But what do you think are the biggest challenges, or do you think it's just relatively straightforward to adapt your method? In particular, I would be interested myself.
For the implementation of the API?
Yes, correct.
Right now, we're implementing it on the instrument computer itself, and that can do a lot of pretty heavy computation. So we're using Comet as real-time search, and we have built beta databases with mostly the most complex thing would be phosphorylation, with about 3 modifications per peptide, maybe 3 phosphorylations per peptide, and the search times there are still pretty quick. So we did upgrade the RAM. So we upgraded the memory on the instrument computers that come from Thermo, but they're pretty good computers. Now, if we want to do something more complex, like go into peptidomics, like immunopeptidomics, something that's non-tryptic, where we have to do non-specific searches, I think that would be a little bit more difficult. It might need more computing power outside of what the system has.
Mm-hmm. Thank you. Well, maybe I have a follow-up study question also. Yeah, this is just fascinating to think about what this would open up as opportunities. And so I was wondering when, for example, looking at tissue samples where sometimes the dynamic range is much much harder versus in, say, kind of a HeLa cell line, particularly maybe thinking about tissues such as muscle or, I mean, even other tissues are quite challenging. Also, do you see any problems there, or how would you also overcome some of those aspects?
There are some things that you can change in the method, even through Thermo's system right now, where you can actually limit the number of peptides you try to quantify per protein. And other groups have taken that kind of to an extreme, and we have too in our more recent work, where you can ask it, you can even track the number of peptides you quantified for proteins across fractions, if you have fractions. And we have seen that can actually help us go much deeper into the proteome. And it actually helps find some of the samples in proteins that are hard to determine or detect in TMT labeled. Samples where, say, they're only present in 3 of the 16 channels or 3 of the 18 channels. Those are going to be very hard to find because they make up a small percentage of the actual sample. And those changes would be, some of the most interesting. So we found that limiting the number of peptides per proteins can help us find those. The only challenge with that is that we do also then, limit some of our statistical power, because we do tend to use MS stats, which actually uses different peptide measurements within the mathematical model to produce statistical, significance. So there are trade-offs, and we're still trying to figure out what the best way is, but there's definitely nice, more advanced things that you could do to try to find those really low abundant proteins you might miss, especially with an MS3 type of method.
Yeah, cool. Ben?
Yeah, I kind of it's almost a follow-up on what you're saying, and this is just my ignorance. I mean, is this, aside from single cell, do you apply the same sort of methods to other things like CSF or like unenriched phospho or things like, I mean, do you want to talk about these not single cell applications?
Yeah, we do. In our lab, we actually use the real-time search strategies and we tend to use the one that comes with the Thermo's instrument. We use that for any TMT experiment. Now, we have other methods with the API, which, I've been editing the code so that we can at least make the code that actually does the method part and then also the data analysis part in real time open source, although the parts that interact with Thermo's software can't be released open source, but we're going to separate them enough, I think, to show people what we're doing. And for that, we are using these basically for almost every sample we have. And when we have those more complex ones where we want to really dig deep into samples, try to go to the very low level ones, then we have been using the API for that. Yeah. And we have used it on things like CSF, potentially even more complex things like, things that might have more miscleavages or non-specific digests. We're working towards that as well, hopefully.
Great.
Thank you.
Yeah, I mean, I was thinking real-time search could actually apply to other mass spectrometry workflows, right, in general, because you showed quite nicely how you made your analysis quite a lot more efficient.
One of the many people, I think, John Yates likes to say, like, the idea of doing this has been around for a while. I think MS3 was really the best use case for it because the MS3 scans are so slow. And if you want to do them, it really helps to do the ion trap scans and then to trigger the MS3s. And people have used that in single cell now to show that helps. We're exploring other ways of potentially using it. And I think you'll see a lot more methods coming out, particularly around, targeted analysis and things like that, where you might want to more intelligently select when you're actually going after peptides. So I think I do think that it's going to become more common, these types of methods.
Great. Yeah. Chris, thanks again for coming in and talking to us again. I'm sure we'll all see you at ASMS in like a month or two, right?
Thanks.
Great. Yeah, thanks. And so next up is David Roberts. He's going to be talking about, well, I'll let him introduce it, but these are probably two of the hardest things I know about from University of Wisconsin in Madison. So take it away, David. Oh, and again, just a reminder to everyone before we go, use the Q&A and then we'll do your questions. Thanks, David.
No problem. Thanks, Ben, for the kind introduction. Can everyone see my slides and everything?
Yes.
I didn't pay for it, right?
Great.
On the receipt, it's not showing.
Okay, so with that, I'll get started. So to begin, thanks everyone again for the wonderful session. I think I'm really happy to be with everyone again, even virtually, on this Best of US HUPO series. So today my talk will focus on, and it's kind of wordy, but it's elucidating the structural variations and O-glycoform heterogeneity of SARS-CoV-2 variants by top-down mass spectrometry. And I'd like to start by kind of giving first some idea, or like at least establishing the framework of why we're interested in doing it this way. So one of the important things that I stress, and I think a lot of people in modern proteomics starts to stress, is the importance of not just looking at intact proteins or looking at proteins in general, but to look more specifically at proteoforms and their relevance in biology. And so what I'm showing here is kind of a cartoon illustration, first of all, to lay the idea of DNA going to RNA, going to proteins, kind of the classical central dogma. But this figure shows a little bit of a rehash of that, where instead of just focusing on everything going in one linear flow, you can actually see that as you incorporate potential for mutations, alternative splicing, and different isoforms, you actually can lead on top of the presence of different post-translational modifications to a diversity of different forms of the same protein, all arising from the same gene. And truthfully, if you track the flow of information between the genome, transcriptome, and proteome, you can actually start to see this exponential increase in information as a result of this. And so sort of with the success of the Human Genome Project that occurred 20 years ago, and as you can see here with this kind of illustration of increasing exponential complexity, you really start to get an appreciation that the proteome itself is extremely diverse and encapsulates all sorts of hidden complexity you would not be able to achieve at what I'm calling here at the genome level, which is what I think of as a static blueprint. But the proteome itself, I really want to emphasize, is kind of this living, dynamic, real life building blocks of biology. And in particular, our lab focuses on using top-down, which is a powerful and disruptive technique that is capable of analyzing proteins at this proteoform level. And it happens to be a particularly useful thing because when you want to look at post-translational modifications, specifically related to COVID proteins, ACE2 and the S protein for SARS, you actually start to gain an appreciation of why this is very important. And so to give you some idea of this, if you look at just the kind of sequence homology at the basic level between the current SARS-CoV-2 versus the earlier 2002 SARS-CoV-1, there's actually a tremendous amount of sequence homology between them, both at genomic and as well as proteomic level. And that doesn't necessarily capture the full complexity of what we experience at the true pathology, right? The same drugs don't work, the same sorts of treatments don't seem to be doing the same tricks. So there's a lot of things that are hidden, not just at this sequence level, that we kind of need to get a better idea of. And one of the groups in particular, Max Crispin's group, have done a great job at revealing that the big and most dominant form of these discrepancies that we tend to see with these viral proteins, especially related to COVID, are the fact that they are dominated by glycosylation. And you can see here this kind of overview of the various nanostructures that you can achieve just from the S protein alone. And following up that, Romy Amaro's group at UC San Diego have done a great job in kind of showing some of the molecular modeling to highlight the important roles of glycan in viral structure. And this is a combination of a view between the S protein and the ACE2 host receptor protein, you can see here just these blue dots that are kind of decorated along these protein surfaces. This is kind of an overlay of different glycan structures that have been identified on top of some protein surface mapping. And it really does look like a ginormous mess, right? It's like a huge jungle of glycans. And so hopefully you can see like this really sort of dictates some of the functional relevance of this disease. And it's not really a surprise either, because functional roles of glycosylation have been established across a variety of in viral biology, ranging from attachment entry, adaptive immunity, spread innate immunity, and this is some of the major key features in assembly and exit. So HIV is a very key player, for example, Zika and so forth, that have shown that glycosylation, both at the virus and at the host level, seem to be playing major roles in how to understand their functional roles and how to treat them. And I want to highlight this. Actually, Lance Wells gave a great talk at the in-person US HUPO 2022 seminar earlier, where he showed some of this work that he did in collaboration with Robert Woods' group. And they revealed that glycans actually have critical influence on the viral receptor interaction of ACE and S protein. So what you're seeing here is not just a molecular model of the protein surface with glycans. They actually showed with data and combination of molecular modeling that there are already glycan interfaces that occur and kind of happen at this ACE-S protein surface. So really highlighting and kind of underscoring the need for the molecular characterization of these glycan structures of this and other emergent SARS variants, also to understand how they are behaving, how they are interacting. And so from scientific point of view, that presents a couple things. So one of them is what, well, I'm calling this motivation and rather exciting opportunities. So at the core, we understand that protein post-translational modifications, in this particular case, mostly glycans, are heavily implicated in viral function, viral structure, and it's crucial that we decipher them. This is a particular challenge because if you want to solely rely on the traditional methods for characterizing protein interactions or protein surface interactions, those are like X-ray, cryo-EM, and so forth. They're very useful for trying to understand how certain residues might be interacting, but intrinsically, they're not completely amenable or totally amenable for a comprehensive glycoprotein characterization. And that's because you have this inherent flexibility of the oligosaccharides. They're dangling and there's all sorts of heterogeneity. And unfortunately, there's no one crystal to rule them all in this case. But that's where we see the potential for top-down mass spectrometry, which holds kind of enormous, what I call potential to provide a proteoform resolved and structurally detailed answer to these significant questions. And so what I mean by that is the hope is that you can use top-down MS to give yourself sort of this bird's eye view of complex proteins. And this is something that our lab has shown before. So in a previous work I've done with my colleague, now graduated Tim Tianbang, we showed that you can achieve high resolution molecular fingerprints of diverse cardiac troponin I proteoforms, which is a key biomarker in heart disease, to establish proteoform pathology relationships in complex samples from tissue and from blood. And more recently, I'd worked on developing a kind of a new approach to look at glycoform heterogeneity from the top-down point of view, mostly focusing on O-glycosylation, as it's a rather difficult type of glycan to study by conventional approaches. And so the hope here is that we can use top-down MS to enable both the simultaneous characterization of these structures, the site specificity, and potentially even the relative abundance of these various glycoforms. And so I want to show you kind of how that is done. So I'm calling this a hybrid top-down mass spectrometry approach for glycoprotein analysis, and it takes place by leveraging two very important technologies. And the first is the Fourier transform ion cyclotron resonance MS. So this is an ultra-high resolution mass spectrometer capable of great and in-depth detail of different proteoforms at high resolving power, high resolution, to really be able to accurately quantify different PTMs. And the second is timsTOF, which is kind of a recently emergent technology with a front end ion mobility, trapped ion mobility capability at the source. So you can get this orthogonal ion mobility information for glycoform structural heterogeneity analysis on top of the combination with the PTM analysis. And to give you an idea of how that might look, here is kind of a very detailed view of how you can do this for the S protein, and particularly focusing on the regional binding domain natively, where we take the timsTOF and we separate different glycoforms first at the front end, where we can take monomeric versions of different glycan structures, observe their ion mobility natively, and then subject that to further fragmentation, where we can start to characterize the various glycan structures. And you can see here, if we take this intact sort of native top-down approach, we can get very well resolved, high detailed MSMS spectra, rich with both the glycostructure as well as the assignments of the site. And very kind of cool with this trapped ion mobility approach is you can actually visualize some of the structural heterogeneity that some of these glycans imbue to the protein. So even just this figure here, you can see that different core 2 type O glycan structures tend to enhance certain features of the ion mobility of these protein ions as they're flying. And you can actually demonstrate that there's an enhancement locally of different collision cross-section values related to different glycostructures. So that gives you some idea of the heterogeneity landscape. And what I think is probably one of the more coolest things that you can do with this approach is this idea of very proteoform specific O-glycoform analysis. And so this is using the FTICRMS right now. Here's kind of a big survey view of the MS1 of the SRBD with the O-glycans intact. If you zoom in very closely, you can actually see that all of these different features here representing a difference in O-glycoform heterogeneity, you can see there's a diversity of different forms, are baseline resolved. And this is important because now we can actually take each individual form of this, subjected to a specific proteoform isolation with specific quadrupole analysis, and then perform tandem MS using a combination of collision activated association and ECD fragmentation to generate this sort of rich, high resolution mapping of all of these glycoforms. And this kind of illustrates what I think is a very unique strength to this top-down approach, where you're able to simultaneously inspect not just the individual proteoforms, but you're able to isolate them away from all the noise. And you can really get this very detailed, high accuracy characterization of these heterogeneous glycoproteins. And this has been very useful for us for looking into some of these very complicated glycoproteins. And in particular, we thought we could try to leverage some of this approach to looking at some of the emergent SARS-CoV-2 variants of concern. And so as we might be familiar, the original wild type variant for this protein is still ongoing, but now it's also been complicated by further and further mutations related to Delta and Omicron. And I'm just showing here a little bit of an architectural idea of how things have progressed with the rapid emergence of these more transmissible variants. In particular, there are significant mutations in the S protein and specifically to the regional binding domain. And this may be indicative of certain changes to the structure that may coincide with some of the pathology changes we're seeing. And so with that, we kind of want to be able to apply this top-down MS method to give some molecular elucidation to understand some of the structural changes we're getting between these different variants. One sec. And so we're focusing on these 2 new variants, Delta and Omicron, and we're going to see if we can take this hybrid approach and apply it to understand some of these structural changes. And so what I want to first show is this comparison between the wild type Delta and Omicron variants. And you can actually see them all kind of in the survey approach one at a time. And this is FTICRMS data on the left, where you can already see that there's a specific enhancement of different O-glycoforms between these 3 different variants, where the Delta and wild type look somewhat similar, but Omicron, probably given to its high number of mutations, shows a very big diversity change of its O-glycoform presence. The capability of utilizing trapped ion mobility on top of this FTICRMS approach gives us a kind of a unique angle to also isolate individual O-glycoforms. So this particularly I'm showing here on the right is a TIMS MS isolation of a specific O-glycan structure where we take it first from MS1 level, so this is the isolated spectra, and then we can characterize based on MSMS and assign individual neutral loss patterns so that we can put together some of the original structure related to this. And you can see this is a GalNAc-Gal dineuronic acid motif. And it's really nice because when you apply sufficiently soft TIM cell parameters, you kind of avoid some of the challenges with fragmentation of glycan structures that could happen at the source or so forth. And kind of using this, we can actually go further and probe a variety of these different structures to kind of show this very large, high resolution survey view of all of the different glycan structures across the 3 variants. And so this kind of, I hope that as you're seeing this, seems to fulfill the promise of giving a bird's eye view of all the glycoforms with the ability to simultaneously characterize their molecular abundance. And so you can see here between the wild type Delta and Omicron variants, and this is work that we currently have submitted in preprint and in review currently, that you can see these differences in O-glycan structures between the various species. But importantly, because we're doing this from an intact protein point of view, we can actually also quantify the relative O-glycoform abundance between the various core type structures. And I'm showing you here between core 1 and core 2 O-glycan type structures that were found on the S protein regional binding domain. You can actually see a significant enhancement in the core 2 type presences on the Omicron variant kind of uniquely. So for some reason, Omicron seems to have a specific enhancement for those types of glycan structures, but this is something that we can confidently characterize using this intact protein MS approach. And something else that we had also found that's quite interesting is that when analyzing some of the Omicron variants in particular, we were able to isolate what we believe is a novel O-glycosite kind of unique to the Omicron variant. And I'm kind of going to walk you through a little bit of this, where in this panel on the left, you actually see the classic thurine 323. So this is a known, well-characterized O-glycosite that we and others have found for the wild type S protein RBD. It's one of the major O-glycosites that are currently being looked at. But in particular, if you look at the Omicron variant, we actually found that there may in fact be this new thurine 376 O-glycosite that actually is N+3 away from a modified proline residue that only occurs on the Omicron variant. And there's for those of you who realize the glycobiology, the proline interaction with S and T seems to enhance the presence of O-glycan structures or enhance the frequency of O-glycosylation. That's something we may be noticing as well here. Here is an example of the MSMS data between the wild type and Delta showing the presence of the thurine 323 O-glycosite. But for Omicron, we have this kind of simultaneous abundance of the thurine 323 O-glycosite as well as this new thurine 376 O-glycosite. We can actually characterize it with and without. And the relative abundance of this is although rather on the lower end, it's mostly still thurine 323. This 376, though, does seem to appear with a lower abundance, although the frequency of it, although not more minor, is indeed what we think is a new O-glycosite unique to this Omicron variant. And so I hope this communicates that this hybrid approach is a powerful approach for the comprehensive analysis of these molecular signatures, and likely I hope can also be applied to other O-glycoproteins in the future, and something that I'm currently working on. And so with that, I'd like to thank a few people. First of all, my dual mentors, Professors Songjin and Yinga, for always giving me the continuous support and always pushing me to go further with this. And they've been very helpful, especially my collaborators, Dr. Alan Brazier, as well as a virologist in UW-Madison, who we collaborate with deeply. Him and his graduate student Morgan are great. And I'd like to thank my funding sources, the American Heart Association, for providing me the predoctoral fellowship that funds this research. And especially the fact that I'm a part of 2 research labs really does feel like you're part of one large academic family. And so I really want to highlight both of them as quite large groups. As you can see between the Go Lab and the Jin group, But we also like to have fun on top of all the research. So that's what I'm trying to show here. With that, I'd like to say thank you so much for listening, and feel free to reach out to me and the rest of the group at any time. I'm highlighting a little bit of some of the Twitter handles between the group. These are student run in particular. So please support the students as well as any of the research you might be seeing. And thank you so much for listening.
Oh, thank you. This was so wonderful, David. Really inspiring. I think it really highlights again how important it is to look at comprehensive pictures and at all the heterogeneity. And I have maybe two questions, but I'll start with one. So I'm actually quite interested in, so when you compare, for example, the Omicron versus Delta or even other strains, so you pointed out certain like residues where you see more or less of the glycosylation, but I'm also interested in the glycan structures. So do you see very big differences in shifting maybe towards some of the sugars such as sialic acid, or have you looked at the sugar part also, or what do you think? I mean, it's such a hard question, right, to know what contributes.
Absolutely. Yeah, so that's a great question, Birgit. I can say, so for the Omicron variant in particular, it does seem to have an enhancement for the sialic acid type glycostructures. This is something that's very difficult to analyze just in general because they're so labile. It's really hard to know if like you're always getting them. There's a lot of technical things that go into that analysis, like trying to be as soft as you humanly can in a mass spectrometer to not blow up everything. But then when it comes to looking at the glycostructures themselves, there is a little bit of redundancy in some of the O-glyco variants that we see in terms of the structural roles that they have. So the certain types of structures seem to be kind of appearing all the time between the different variants, but just in different molecular abundances. The exact stereo and enantiomeric versions of these glycans are another question. We're very confident in the assignments, but the fact that we don't know necessarily their stereo makeup per se, because it's a little bit challenging. It's actually something that we thought that using trap dye mobility would help, and actually indeed it does give us more confidence when we assign some of the linkages, but it's something that I hadn't investigated, but definitely has mattered before for other viruses. So that I think it's excellent to say. And actually we're now looking into more of the host receptor interaction natively. So we're looking into how this might corroborate with ACE2 at the receptor membrane level, which is very challenging, but also hopefully going to be very interesting when we get there.
Yeah, I can imagine. I have maybe one other question. I mean, we've done a lot of work on not as sophisticated as you, but thinking about glycosugars in cancers, right? And specifically breast cancers. So I could imagine that your technology that you have developed, one could equally apply to looking and asking more about heterogeneity and proteoforms, and for example, the relevance of glycoforms during cancer, such as, or even in the prostate cancer, right? The PSA, it is known that a glycoform is the more detrimental aspect of this. So, I mean, could you see your technology really expand into other areas also?
Yeah, that's a great suggestion, Birgit. And in fact, it's something that I think would be amenable for this. A lot of the focus right now is more so on the cardiovascular side, given the kind of areas that I focus on with my labs. With Ying especially, she's cardiovascular proteomics person. But in terms of cancer research and even sorts of other areas of surface proteins that tend to be heavily glycated, these I think are some of the things that are going to be very enabling, because there's all sorts of difficulties in being very confident in what exactly are the glycostructures, not just the sites, but the glycostructures themselves. And that's something I think, bottom up does a great job of getting you sites, but I think really getting the structures is something that this sort of top down type approach would be very enabling for.
I bet. Yeah, great. Ben?
Yeah, no, excuse me. I'm in such awe of the work that comes out of the group. This top down, it's such a hard problem. I have a really basic question I've always wanted to ask. Yeah, we always talk about how when we were doing 2D gels, which by the way are not good for high throughput, should not be used like this. And I kicking myself for asking, but we always said that there were these like trains and it was, oh, that's glycosylated. Does anyone in your lab, do you make someone like run a 2D gel and then see these things, or y'all are really just staying in the mass spec?
You know, it's funny. I haven't run a 2D gel. I don't think anyone in our lab has run a 2D. Well, so
You shouldn't.
2D.
I mean, I don't
Yeah.
wish that on anyone.
So let me primer. I have run a 2D gel in the past. I've never run it on glycoproteins, or at least when I ran it, I didn't know if it was glycosylated. That was a long time ago. But no one in the lab has done it. But actually, you bring up an interesting thing. Running gels, at least just one-dimensional gels, like after dealing with glycoproteins, you see a very characteristic smear. It's never just one thing. It's the protein band, even just like on its own, like for some of these things, just like take up like thousands of molecular weight differences. So I'm actually curious. I haven't seen it before. Do you see something very particular when you run 2D gels of glycoproteins?
It's yeah, and I can't believe I'm talking about it, but yes, it's you would see things like ApoE, and you would see these, this kind of train. You call them trains, and we would always say like, oh, it was this, but then you take the spot and you chop it up, and you're probably not looking at the glycosylation anyways, and so there was probably more hand waving happening. That was 10 years ago, but
you can
just
actually. So I'll disclose this. So I have actually tried separating proteins on a gel and seeing if you can still do glycoproteins intact. It actually works. It recapitulates really well. So we could take the ancient wisdom, go back to the 2D gel, cut it out, and see like, and then apply it to this really fancy like setup.
And to be fair, I mean, it's, I think the problem was always, what you're doing, like this rapid analysis, like it's more quantitative, it's reproducible, like that's not something that was necessarily possible. But, and that's what makes this, I think, so amazing is, right, you're talking about doing this like at scale, which is, yeah.
I appreciate that. And yeah, it's really humbling too, because you think that so much of this is direct, right? Just because the mass spectrometer tells you what the mass spectrometer tells you. And then you find like even just trying to do intact, there's so many ways that the data can be biased if you're just not careful with how soft you're being with the glycostructures, because everything can collapse into something that looks like something else. Yeah. So there's a lot of challenge with that. I think the native approach really helpful. It's really helpful, but it's definitely something that still needs more technical development before this becomes more widely available. So hopefully in the future, we'll see some advances. So stay tuned.
Yeah, and one more question, if I can. Wait, let's make sure. Okay. This is obviously on a pretty hot topic, but you gave some excellent references. People have been looking at this and other envelope glycoproteins. I mean, do you, and you mentioned you're in a lab that's also doing all of these cardiac markers. I mean, do you find, if you were going to predict the future, like, do you want to kind of keep going down this viral glycoprotein road, or getting more into disease biomarkers where cancer, Birgit was talking about cancer glycosylation?
Ben, so I admit, this is kind of funny. So the path that I actually tend to go down personally when it comes to like science is actually very outside of the glyco world. It's something that I've only kind of recently adopted for myself. And to be honest, it became a thing because during the lockdown, when everybody was, not able to get into a lab, I was losing my mind, and I had very supportive like mentors who were willing to let me try things. So I go out and I write a grant for the AHA, and they buy it, and then now I'm allowed to go back into a lab. So a lot of this work I'd done in just like a year, and just like being alone with the instruments in like not a dark room, but like, it gives you a new way to look at things. And so when I now see the opportunity for this, at least especially when it comes to cardiac, is that I'm seeing an opportunity for this in terms of membrane receptor proteins. So that's actually something that I'm working on very much. And cancer membrane receptor proteins matters tremendously, because so much of the surface in cancer and other disease biomarkers is like what is actually changing and what you can actually probe very effectively. So I do have a lot of plans to look into not just viral glycobiology, but looking into some of these more disease biomarkers that are critically implicated with glycosylation as a pathway.
Cool.
I just really love what you just said, David, and I think you are the perfect example for finding something really positive in something so tragic as the pandemic, right? And making the best out of it. And I think that has that's really wonderful. With your creativity, you generated this new, advancement for you. No, that's really wonderful. Yeah, I agree. Surface proteins are important, right? Also in aging, and definitely sounds great. Thank you very much, Ben.
Yeah.
Thank you so much.
Thanks, David. Right, and as we keep on rolling, again, I want to remind everyone, please use the Q&A if you have any questions. Our next talk is one that I did get to see at HUPO, and I love that I get to see it again. It's Wout Bitchermew is going to be talking about one of his newer tool developments, which does not have a Star Wars acronym. And I just want to formally say again, let's keep it going, Wout. But yeah, take it away, Wout.
Yeah, thanks, Ben. Yes, unfortunately, not a Star Wars reference this time. But yeah, I am Wout Bitchermew. I'm a postdoc at UC San Diego, and I'll be presenting our work on using deep learning to embed spectra and jointly analyze millions and millions of mass spectra. To start off, let's quickly revisit a typical mass spectrometry project lifecycle. First, of course, you go in the lab, or mostly other people, not myself, fortunately, go in the lab and generate data. And as current projects are increasingly ambitious, rather than a few data sets, a few mass spec files, you'll end up with a dozen to hundreds or even thousands of raw files per project. Of course, you need some powerful bioinformatics software then to analyze your data, of which there are maybe a few Star Wars themed tools out there. And then you have some good results, you're excited, you want to share it with the scientific community, and you write up a manuscript and submit it for publication. And typically most journals nowadays mandate that you then also deposit your raw data in public data repositories. And there are a few of those. Here at UC San Diego, we maintain the massive repository for proteomics, mass spec data, and GMPS for metabolomics data. However, as soon as the data is in these public repositories, there's actually very limited reuse of the existing data, which is a little bit unfortunate. And even when data gets reused, it's typically only a handful of data sets that get reused by other researchers over and over again, while the vast majority of data that is in these repositories remains underexplored. And this is problematic. Basically, whenever you have data associated with a publication, it is almost as if these different data sets exist in isolation, and there is no, people don't look at data across different projects or even across different repositories. This is unfortunate because, of course, when you see a certain peptide or an analyte in a specific experiment, nowadays, people act as if this is the first time that this peptide was ever seen in the whole history of analytical chemistry, right? But actually, the same analyte might have been observed in a different project as well. And it would be very relevant to have this information available and to then inform your subsequent analyses, because this is super valuable information that you can use. So our idea was that we want to promote data reuse and perform repository scale analysis by embedding MSMS spectra into vectors. And so I'll be talking about two different data types during this presentation. On the one hand, we have MSMS spectra that we are very familiar with. But on the other hand, I also have vectors, which is maybe a little bit of a newer concept. And I'll briefly highlight some differences between these two data types. So spectra, even though we know very well how to interpret this data, when you look at it objectively, it's actually a quite complex data type. It's like multidimensional. It requires specialized algorithms to analyze these data. All of the awesome tools that we have to our availability, but nevertheless, it requires quite some expertise to properly analyze the data. And also, as you're all aware, especially with these newer instruments, they can spit out a huge amount of data for each experiment. So this data has a large memory footprint. In contrast, vectors, which are basically just arrays of floating point numbers, they're a very simple data type. Maybe it's a little bit abstract for you and me, but this is really the bread and butter of what computers use. So this is the most common data type for computers. Because of that, there are also a lot of algorithms available out there. You can just use out of the box to analyze vector data. And vectors have a relatively efficient memory footprint. So our idea was to develop a method which we call gleams to embed spectra into a low-dimensional space. So convert MSMS spectra to vectors with the important characteristic that these low-dimensional embeddings have to be close to each other in this new embedded space if they are generated by the same peptide. And this is our gleams method. And so underlying gleams is a Siamese deep neural network. And I've sketched here how it works, how we train this neural network, and the loss function that we're going to optimize. This may look a little bit intimidating, but I'll walk you through it and you'll see that it's actually quite straightforward. So gleams is a Siamese neural network, and this means that there are two instances of our embedder network, indicated in purple, that are identical. So we have two separate instances of the same embedder network. And each of those two instances of the embedder network will take a separate spectrum as input, spectrum 1 and spectrum 2. And there's a third piece of information that it takes as input during training is the label Y. This label will be 1 if both spectra correspond to the same peptide, and 0 if both input spectra correspond to different peptides. And then the embedder networks take these spectra, convert them to our embedding vectors, as described previously, and the network is trained by optimizing the Euclidean distance between the embeddings for those two spectra. And this is the loss function that we're going to optimize.
Okay, Wout,
sorry?
back. Wout, I think you froze.
Yes.
Or maybe that was me. Oh, please continue.
Yep. Okay, so we're going to optimize this loss function. Now, in the first case, when Y is 1, these two spectra correspond to the same peptide, and the first part of the loss function is active. In this case, we're going to minimize the Euclidean distance between the embeddings for those two spectra. And intuitively, what this means is that those two embeddings will be pulled closer to each other. Similarly, here's another example. The two nodes here are two spectra, and again, the Euclidean distance will be minimized. I mean, they will be pulled closer to each other. In the other case, when the label is 0 and both spectra correspond to different peptides, the bottom part of the loss function will be active. And in this case, we're actually going to maximize the Euclidean distance between the embeddings, and we're going to push apart those two spectrum embeddings. We're not infinitely pushing apart the two embeddings. We have also defined a margin indicated here in red, and we're going to make sure that embeddings for dissimilar peptides are further away from each other than the margin. However, in case again, here we have two peptides, embeddings for spectra that correspond to different peptides. If they are already further away than the margin, we don't need to do anything anymore. But again, intuitively, this will make sure that embeddings for similar peptides are close to each other, and that they are, and embeddings for different peptides are further away from each other. Now, how did we train and evaluate gleams? We used a very large data set called the massive KB data set. We didn't have to compile this ourselves. This was already produced a few years ago by colleagues at UCSD. And this is a consistent reanalysis of more than 31 terabytes of public human HCD data available on Massive. As training data, we extracted 30 million high-quality spectra from this very large data set. The spectra that could be identified with the highest confidence, so they had very high-quality training data. And if you recall, of course, Gleams doesn't take a single spectrum as input, but it takes pairs of spectra as input. So we generated all possible pairs out of those 30 million spectra that are within a 20 ppm precursor tolerance window of each other. And this resulted in 541 million positive pairs, spectra that correspond to the same peptide, and 11 billion negative pairs of spectra that correspond to different peptides. And then as test data, we used the entire massive KB data set, 670 million spectra. And for your reference, out of those 670 million spectra, 28% of them were initially identified in the massive KB work that was using MSGF+. But this also demonstrates that there are still a lot of spectra, hundreds of millions of spectra in this data set alone that were not explored so far. The first thing that we did then was evaluate whether training of our deep neural network was actually successful. And again, a reminder, Gleams takes pairs of spectra as input, and these are positive pairs if the spectra correspond to the same peptide, and negative pairs if the spectra correspond to two different peptides. And we can see that indeed training was successul because positive pairs have a very low distance between the two embeddings, approaching 0, whereas negative pairs have a higher distance, centered more or less around 1. Additionally, there's very limited overlap between the distances between positive pairs and negative pairs. So we can very nicely put a distance threshold and minimize our false discovery rates, a false discovery rate when contrasting positive pairs, distances between positive pairs and negative pairs. So this was pretty encouraging. Additionally, when we explore the embeddings a little bit more, we saw that they captured latent properties of the spectra and of the peptide spectrum matches. Here we have visualized 100,000 embeddings for peptides that frequently occur in the data. And on the left, we have colored the embeddings by precursor charge, and on the right by precursor MZ. And we see that these two properties kind of define the location of the embeddings in this space. For example, here we have a cluster of embeddings with precursor charge 2, which have moderate MZ. Here we have some smaller clusters, precursor charge 2, but higher precursor MZ. Here a cluster of spectra with a higher precursor charge, etc. And besides these two obvious properties, the embeddings captures some additional properties such as peptide length, what the C-terminal amino acid was, etc. And the nice thing, the exciting thing was that the neural network was able to extract this information and learn these patterns from the data directly. This was not provided by a student network in any way. Just by seeing a lot of data, the neural network kind of learned to find these patterns in these spectra and peptide spectrum matches. As a next step, we then applied gleams to the analysis of our entire massive KB data set, consisting of 670 million spectra. And then we especially wanted to analyze the dark proteome, which are spectra that are repeatedly observed, but consistently remain unidentified. And you can see here again in the initial results, 28% or 185 million spectra could be identified using the MSGF plus annotation pipeline used in the original massive KB work. When using gleams, we almost double the number of spectra that could be identified, indicated in blue. And we do this by propagating peptide labels to embeddings that are close in this embedded space, in the gleam space. So we get a very impressive boost in the number of spectrum identifications over the state of the art. Additionally, in yellow, these are more than 200 million spectra that were not identified, unfortunately, but we could actually cluster them in the gleam space. This indicates that these spectra were repeatedly observed across multiple projects in our massive data set. And even although we don't know which peptide these spectra correspond to, the fact that they occur repeatedly indicates that it's not just some noise, but that's probably a true signal. And thus these 200 million spectra are a very interesting data set to explore with newly developed algorithms and bioinformatics tools. And we have exported them for the community to investigate this data in more detail. Furthermore, a big advantage of the Gleams approach is that we're now working in this embedded space where we can very efficiently perform advanced analyses. And part of what we did to get these additional spectrum identifications is that we use open modification searching. And very briefly, open modification searching works by using a very large precursor mass window. So you're not only looking for directly matching peptides with an identical precursor mass as the spectrum, but you're also allowing peptide to spectrum matches with large precursor mass differences that then correspond to post-translational modifications. And here on the right, you can see a histogram of the observed precursor mass differences that indicate then which modifications are present. Now, of course, when you're using such a large precursor mass window, your search space really explodes. So performing this at the repository scale, even though there are now some tools that can do open modification searching quite efficiently, this would still be a computationally very challenging task. In contrast, using Gleams, we get a significant data reduction, and then we can actually, it's very feasible to do open modification searching. And you can actually see from these results several common modifications that frequently occur, including some that you expect to see during sample processing, but also, for example, some biologically relevant modifications, including phosphorylation. So to conclude, I have introduced here Gleams, which is a deep neural network that was trained on hundreds of millions of MSMS spectra, and that has learned to capture spectral properties of these data. Using Gleams, we were able to increase the spectrum annotation rate by 71% over the state of the art, corresponding to hundreds of millions of newly identified spectra. And this has also enabled us to discover many peptides with post-translational modifications. And this kind of shows that there's kind of a little bit of a phase transition when going from individual experiments to the repository scale, because a specific spectrum might remain unidentified and unknown in the context of an individual experiment. But if you look at the spectrum then in the context of an entire data repository, you might find some matches to other spectra that are highly similar to it. And this, scaling up to hundreds of millions of spectra was really only possible using the GLEAMS methodology, whereas other existing tools were not able to operate with such a massive amount of data. And we're actually currently exploring extending GLEAMS even further to billions of mass spectra, just like churn all of the mass spec data that we have on Massive Pride, etc., put it all through GLEAMS. We're working on some incorporating some novel deep learning architectures to achieve that, updating our training procedure and so on. But this is very exciting to further extend GLEAMS to basically all mass spec data that is out there. To conclude, I want to thank all of my awesome colleagues at the University of Washington and at UC San Diego. And then I especially want to call out Damon May at UW, who started the GLEAMS project with me a few years ago already at this point. And then also Bill Noble at University of Washington and Peter Dorestein at UC San Diego, my two mentors who have always supported me in performing this research. Finally, GLEAMS is publicly available on GitHub with instructions to try it. So if you're interested in it, give it a whirl. If you have any questions, please reach out to me. More information is available in currently still a preprint, but our work is also in press at Nature Methods, so hopefully it will become available through there in the near future as well. And with that, I'm happy to answer questions.
All right, well, Wout, that was very inspiring. Thank you. Yeah, the scale of your projects is very high. And so I had a question. So for example, your vectors, can they take into considerations, for example, aspects like ion mobility? But then also my second question that goes maybe into similar directions, can you, when you have post-translational modifications, does that bring in a different level of complexity for you, and how do you handle with that? And is that different for different types of PTMs?
Currently, we don't consider additional information such as ion mobility, but also even retention time, etc. We don't include that in as part of the input to our network yet. We just use individual MSMS spectra. And then the second part is, the second question was, do PTMs, can they cause potential issues? And I would say no, to a large extent. Our methods, the input of GLEAMS just takes spectra and peptide labels as input. Whether those labels, those peptide labels include post-translational modifications is up to the prior spectrum identification step. That's also because you saw such a large number of phosphorylated peptides in this plot, which wouldn't really it's not a modification that you would see that often compared to the other ones in a typical data set. The problem here, of course, is why did we see so many, is that in the original massive KB reanalysis, the team had not specified phosphorylation as a variable PTM. And so no phosphorylated spectra were included in the training data for GLEAMS. But of course, on massive, there are a lot of phospho enrichment studies, which could then be subsequently identified using GLEAMS. One of the other evaluations that we did is then GLEAMS didn't see any phosphorylated spectra as part of its training. So this was actually a nice test for us to evaluate the robustness towards unseen data and data with different characteristics. And it was very encouraging to see then actually that the strong performance of gleams was retained even for those phosphorylated spectra, even though it had never encountered that during training.
Great. We have some questions actually that I'm going to just read. But first off, again, I love seeing your progression, and I'm glad to see that you haven't been taken over by the metabolomics people. First question about the vectorization of spectra. There's different spectra vectorization algorithms in the literature. Which algorithm is used in gleams? And are there pros and cons over using one or the other?
Yes, there are definitely pros and cons. Actually, the unique aspect of gleams is that it's a learned embedding. The neural network, it's kind of a black box. Spectra goes in, and then a vector comes out, and this vector is only 32 dimensional. So it's 32 floating point numbers to represent a spectrum. So it's a big condensation of information to go from a spectrum that can have hundreds of peaks down to just 32 numbers. But this is kind of the transformation that the neural network has learned. And even though these vectors are so short, they're very information rich. As I showed, they capture fragment information, they capture precursor information, they capture even some peptide label information, even though that was not explicitly provided during training, etc. So I think that this is really an important advance in being able to vectorize spectra. And then now, as I mentioned, also there are a lot of off-the-shelf vector algorithms out there. Now we can actually convert spectra to low-dimensional vectors, use these existing algorithms from the data science world and apply them to our mass spec data.
That's great. Another question, and I'm going to read this one more so I get it right. Can you use gleams to submit de novo identified tandem mass spec data and screen for hybrid or splice peptides, which derive from fusion of different sequences from different proteins?
Yes, you can. So even though gleams requires peptide labels during training to figure out whether these are spectra correspond to the same peptide or not, during inference, so after gleams has been trained, you don't need peptide labels. So it can be spectra identified through sequence database searching. It can be spectra identified using de novo. It can be unidentified spectra. Peptide label information is no longer needed once the model has been trained. So what you could do is you could, if you have some of these de novo identified MSMS spectra, and you know that they correspond to splice peptides, or similarly some exotic peptides, you can embed them. And then if you have unknown spectra, if you see that these spectra are close in the embedded space to some of your peptides of interest, you know that they are very likely they are similar or they have some related characteristics. So that is definitely something that you would be able to do with gleams.
And that almost, and I'm sure you get asked this a lot, is the application to these immunopeptidomics data sets, because I feel like that's obviously a pretty hot topic in a hard search space.
Yes, indeed. I think again that this is a very interesting potential application for gleams, because indeed in immunopeptidomics, the search space is so big, and there an approach like gleams can help to again reduce the data, condense your large amounts of data to the most salient pieces of information, and then more efficiently try to identify these immunopeptides and other types of data.
That's great. And I just want to point out, all of, in addition to this video, but all of your talks and the papers, you're very good. So if anyone also has more questions, they can see that. You're on Twitter and other things, right? Whoop.
Exactly. Yes. I will put the slides up on Zenodo later today as well, so that everyone can find them. And if you have any more questions, chat me here, or I put my email and my Twitter handle up in the presentation earlier as well. Reach out to me afterwards. Happy to discuss some more.
Great. And thanks again for coming. So
Thanks,
next
Ben.
up, so next up is Ruth Huttenheim, and I'll let her introduce the title. But again, this is, I want to again point out, please use the Q&A. And yeah, thank you. Ruth, take it away.
Awesome. Thank you so much. Thanks for organizing this event and obviously inviting me to present. I'm very excited. Hopefully you can see my slides in the right mode. So I'm going to talk about visualizing spatial temporal resolved interaction networks of G protein coupled receptors in pain. As you all know, the new opioid receptor is a major target for pain management. But the problem is with opioids that target these receptors, they're highly lethal. They are lethal in overdose, they're highly addictive, and their prolonged use leads to tolerance. So there's an urgent need for safer analgesics. And you can imagine that, and there are different approaches taken so far towards safer analgesics. On the one hand, it is to develop novel agonists that target exactly these receptors, but maybe with distinct signaling properties, so that sorry, I will just change the pointer options. Yeah. So that you might preserve the analgesic effect, but have less side effects. And there are many efforts right now taken in this direction. The other way that you can imagine novel pain medication is to target other receptors in pain transmission. And I don't want to read all in details what is shown here on the right side, but I want to emphasize that along the pain pathway that you see here, from the periphery to the spinal cord, there are a large number of GPCRs that are expressed on the way, and they have already shown to impact this transmission. But what I want to argue before we actually attempt to design this safe analgesic, what we need is first to understand the mechanism, how these GPCRs signal and how their signaling is regulated. But also we need a method then to assess how these novel molecules that we design activate the receptors. And so because I'm talking about this a lot, I want to give you a brief introduction in GPCR signaling, which is quite complex. So what you see here is a receptor that's activated with a ligand, and in the first pass after activation of the receptor, it initiates the G protein dependent signaling by engaging the heterodimeric G proteins based on alpha and the beta gamma subunits. And these then dissociate into the alpha and beta gamma subunits individually and activate downstream effects such as activation and inhibition of cyclic AMP or opening and closing of ion channels. Then the G protein dependent signaling is terminated by phosphorylation of the receptor, here the G protein receptor kinases. And then the phosphorylated receptor recruits arrestin, which then leads to internalization of the receptor and trafficking to internal compartments. What's really important is that over the recent years, what has emerged is that the receptors can also signal from internal compartments and not only from the plasma membrane, so adding to the complexity of receptor signaling. And then after endosomal sorting, these receptors are then either recycled back to the plasma membrane or degraded in the lysosome. So I hope what you can see from here is that there's a lot of regulatory events going on to activate the receptor. And this has been very traditionally difficult to capture. And one reason, because these are membrane proteins, and right, we know that biochemically they are difficult to study. Most of the interactions that are happening here of transient nature, they happen over short time points. And what I wanted to point out here is that, the receptors changes cellular location. And this is really important because signaling from internal compartments can have a different function than from the plasma membrane. And so in order to capture all of these events, we need a method that can capture the interactions of GPCRs with temporal and spatial resolution. And so a few years back, we attempted to develop such an approach based on proximity labeling, utilizing an enzyme called APEX. APEX is this engineered peroxidase, and just to briefly explain how it works is that when you express these APEX enzyme in cells, treat the cells with biotin phenol and give a short pulse of hydrogen peroxide, APEX turns biotin phenol into biotin phenoxy radicals, and these are highly reactive and they label proteins in proximity with a certain radius. And so we tested this then, or we developed this approach for our GPCRs in a way that we fuse the apex enzymes to our receptor of interest, and we express this fusion construct in cells. And what we can show is that if we perform the biotinylation reaction after certain time points of receptor activation, we can take this snapshot of the proximal proteome around the receptors at these given time points. And so the nice thing is now you capture these in intact cells, you can then purify the biotinylated proteins using harsh lysis conditions, and then quantify the biotinylated proteins over the time course. And what we showed in this publication a few years back is that we can identify the proteins that are engaged by the receptor, how these are changing over the time course of receptor activation. But in addition, because we have a certain labeling radius, we can obtain some spatial information about the receptor. And I want to talk about this a bit in the next slides. So having this technology, we thought, well, can we apply this to the mu-opioid receptor and see how diverse is the proximal proteome of the receptor when we activate it with diverse ligands. And so to do that, again, we fused, we generated these apex fusion construct for the mu-opioid receptor, generated stable hex cells, and then we chose 3 very distinct ligands: DEMGO, a peptide ligand; morphine, which is used as an analgesic in the clinic; and PSEN21 is a very novel ligand designed by with novel chemistry. And so what I can say for these 3 ligands is that they are known to be different in terms of the cellular response. And we look here on the left on G protein activation, on the right on beta-arrestin recruitment. What you can hopefully see is that while all of them activate G protein signaling, there's a clear difference in terms of beta-arrestin recruitment, where DAMGO shows strong recruitment, morphine a little, and PSEN21 has negligible recruitment of beta-arrestin. So we wanted to understand how diverse is the proximal proteome of the mu-opioid receptor when we activate with the 3 ligands over a time course of 60 minutes. And we took this biotinylation snapshot at different time points that you see indicated here. And so the first question that we want to ask is, what are overall the proteins that change in biotinylation over the time course? What we see here now depicted in the heat map are proteins that significantly change in biotinylation for at least one of the drugs. And I hope what you can see is that the strongest changes we see for DEMGO, less for morphine, and not much changes for PSEN21. And when we cluster these, they form distinct clusters, mainly in proteins that upregulate over the time course and downregulate over the time course. And we were wondering, well, what are the proteins that are represented in these clusters? So now when we perform the gene ontology enrichment analysis, what we clearly saw is that most of these proteins provide information about the localization of the receptor, because the proteins here that are strongly upregulated when we activate the receptor with DAMGO show enrichment for endosomal, lysosomal, and Golgi localization, while proteins that are downregulated over the time course show enrichment for plasma membrane location. So what we can say here from this data is that now the structurally distinct ligands that we used here have strongly influence the subcellular location of the receptor. And I told you that's important because these receptors can signal from different locations in the cell. But also, I think one part is that it's clear that the apex data can capture both the spatial effects, so it can capture spatial specific proteins, and somewhere, believe me, in this data set, there are the interactors that regulate receptor function. And so the question is there a way to dissect these 2 temporal profiles of the interactors and the spatial bystanders? And to do so, we developed a computational framework that relies on spatial references. And these spatial references is just like the apex enzyme fused to protein localization domains to target them to certain compartments. And we sourced 3 compartments here: plasma membrane, endosome, and lysosome, because they are relevant for receptor function. Now, what we can do with these spatial references is that we can estimate the background labeling at certain compartments in the cell, but then we hopefully can also use this information to estimate receptor localization. So to start estimating receptor localization, we thought, well, first we need proteins that can indicate receptor localization. And so we looked for, by pairwise comparison of these spatial references, the biotinylated proteins of these spatial references, we looked at which proteins are overrepresented for certain compartments in the cell. And now, and these are depicted here in the different colors, and we call them our location-specific indicator proteins. Now, with these indicator proteins, we have the intensity over our spatial references, but also we have these proteins quantified in our receptor data set. So what we then thought is that we can determine, we can predict the drug and time-dependent receptor localization based on these location-specific proteins, and basically by solving this linear equation here. So we thought it's like, what is the combination of our endosome, lysosome, and plasma membrane intensities that can predict the intensities that we measure for a receptor for when it's activated with a different drug at different time points. And so we then determine basically a coefficient for endosome, lysosome, and plasma membrane for each drug and each time point. And so we can, when we then plot these coefficients for each location and each drug over the time course, we can see beautifully the trajectory of our receptor over time. Just to show that here, when we look at the receptor activated with DAMGO, we see a decrease in plasma membrane location, increase in endosome and lysosome localization. And so see the trafficking pattern of the receptor. However, in strong contrast, when we see when the receptor is activated with P721, there's no change in the localization of the receptor. So this is pretty cool. Now we have a way to predict receptor location based on hundreds of proteins that are biotinylated over the time course. But what we can also do now with these coefficients that we measure, we can estimate for all the proteins in our data set, well, what is the expected intensity at each time point in each drug, based on the localization change of the receptor. And so just when we have this expected intensity, we can compare to our observed intensity and subtract it. So I want to visualize that based on some examples. So one of these is an endosomal proteins, EA1, which we do not expect to interact with or function with the receptor, but it's labeled based on the receptor moving through the cell. So now we see this increase in intensity because when the receptor is activated with DAMGO, because it undergoes trafficking and goes to the endosome. And exactly the same pattern we see when we predict the intensity of EA1 using our location-specific coefficients. And so when we now subtract the observed from the expected intensity, we see that in our detrended data set, there's no change in intensity anymore. So we could predict the localization change of that receptor over time. So now when we look at arrestin, which I told you before, that's a known interactor, we see this strong change in biotinylation for when the receptor is activated, DAMGO and morphine. But when we calculate the expected biotinylation pattern or the intensity of arrestin, we would not expect this based on that arrestin is labeled based on the localization change of the receptor. So now when we subtract this, right, we preserve still these intensity pattern because arrestin is actively recruited to the receptor to function with it. And so now you can imagine we can do this for the whole data set and account for the spatial location change of the receptor, and therefore determine which proteins show a change in intensity that is by active recruitment to the receptor. And so now you see here another heat map that depicts the remaining proteins that show still a significant abundance change for in our detrended data set. Again, most of the changes we see still for DAMGO, less for morphine, and when the receptor is activated with P721, changes are, there are not many changes. And so one of the examples is again, arrestin that I showed you before, which shows these differences when the receptor is activated with the 3 ligands. But to our surprise, while we see this huge difference between proximity labeling of proteins, we also found that 2 proteins, EY4 and KCD12, show similar labeling patterns, or at least for KCD12, we see that you see an increase for all of the 3 when we activate the receptor with all the 3 ligands. And so we were wondering, well, that's interesting. What could be the function of these 2 proteins with a mu opioid receptor? So what we know for EYF4, it's actually belongs to a family of transcriptional co-activators. These are multiple proteins in this family, and these are recruited to transcriptional complex via the interaction with other DNA binding proteins. But what was found in the early 2000s that EYF2, another member of this family, could interact with constitutively active G-alpha proteins. Now, for that, we found this interesting, right? And then for KCTD12, what we know, it has some function with GPCRs, specifically with the GABA B receptor shown here, that it acts as an auxiliary subunit. And when the receptor is activated, it interacts with the released G beta gamma subunit to uncouple it from ion channels that the G beta gamma subunit opens. So based on this known function, we hypothesized that EYF4 might actually not bind directly to the receptor, but to the G alpha I protein that is released after receptor activation, and therefore have an effect on cyclic AMP production downstream of G alpha I. And then KCTD12, we thought it's based on the known function with the GABA B receptor, that it can also sequester the G beta gamma subunit from the GERK ion channels and therefore modulate the regulation of these ion channels. So we wanted to test this. In the first step, we performed a knockout of EYF4, activated the receptor, and then measured cyclic AMP production with the fluorescent biosensors. And this is done in collaboration with the Van Zastra lab. So what we can hopefully see here is we show here the fluorescence of the cyclic AMP biosensor over the time course. What you can hopefully see here is that when we knock out EYF4, there's a stronger inhibition of cyclic AMP. So there's more activity of the inhibitory G alpha I subunit, and that indicates that EYF4 sequesters active G alpha I to keep it in check. And when it's not there anymore, there's more activity of G alpha I. So for KCTD12, then we also performed a knockout of KCTD12, and we tested then using electrophysiology in collaboration with the Levitz lab at Cornell, how it changes the current that runs through the GERK ion channels. And so what I hope you can see here is then we look here on this plot at the current that runs through these ion channels. And for our control, you see a rapid decrease of the ion current versus when we knock out KCTD12, the ion channel stays open for a longer time, which means indeed that KCTD12, it usually sequesters the beta gamma subunit from the ion channels and then leads to the closure of this ion channel. And now if you knock it out, the ion channel stays open longer. So what I hope it can show you here is that, we have this method based on apex-based proximity labeling that can take a snapshot of the proximal proteome of receptors. Specifically, I showed this on the samples of GPCRs, and we have developed data analysis tool to extract a multitude of information. We can track the cellular location of the receptor, but also map its interaction network, and it's changing over time. So now here I showed you an application to the mu-OB receptor that we activated with different ligands. And we see how these ligands modulate receptor localization of in the cell, but we can also use it to discover novel interactors and or novel, let's say, not direct interactors of our receptor in this case, but proteins in the proximal environment that regulate receptor signaling, in this case KCTD12 and EYF4. And I think the cool thing is now looking at this is also a proteomic-based method that can track receptor trafficking and protein engagement, and it provides a holistic way to investigate novel ligands that target these target GPCRs. So in the last minutes, and I know I don't have much time, but I also wanted to show you another examples how we can use these methods in the highest throughput. And I just show a few snapshots of it. So our goal was now to try to map the pain receptor effector network by selecting a large number of different receptors that are involved in that we know are involved in pain sensing and transmission. And the idea was that, on the one hand, we can use it then if you generate this pain receptor effector network, we can illuminate fundamental receptor biology, but also then in the long term identify putative pain targets. And we selected for this initial study 8 receptors that represent not only different receptor families, but they have also different role in sensing and transmission of pain. And so, well, fast forward, what we needed to do here is first we needed to have a high throughput method to perform this apex reaction and the enrichment of biotranslated proteins. So we automated the whole enrichment process, and we used data independent based quantification of the proteins over time course and our automated data analysis framework that I showed you previously. And so what we can see as one of the snapshots that I want to show you is the effect on the cellular location of the receptor on the different receptors when we activate them with one ligand. And so you see here the 8 receptors depicted and the trajectory through different compartments in the cells. In pink, we depict here plasma membrane. In green, the endosome. Orange is Golgi, and purple is a late endosome and lysosome. And what was really striking to me is, right, when we look at these are all receptors expressed in HEC293 T cells. We activate them with ligands, and they show a large diversity in terms of their spatial organization in the cell. Whereas some receptors just stay at the plasma membrane, like PRX4 or NPFFR1, but other receptors undergo endocytosis and traffic to the endosomes, like PTGR4 and MRGPRX2. So this is really striking to us. At the same time, I told you we predicted then the interaction network of these receptors. And just to show you a last slide of data, give you a snapshot of a complex interaction network that we obtained from these 8 receptors. And what we see here is that, that there's a large of receptor-specific events. These are the color of these nodes depicts like with how many receptors we found this protein interacting or in the proximal environment of the receptors, and in yellow shown proteins that are receptor specific for one receptor, but then there are the increase of the more dark the color of the node is, the higher the number of receptors that we found this protein interacting with. And you see a lot of shared events that represent endosomal transport, receptor-mediated endocytosis, but also other effects like protein kinase activation, actin nucleation events, and so this is now a rich data sets that we are digging into and validating the function of some of these nodes with the different receptors. So last but not least, I know I'm a little bit over time, but I really want to thank people involved in this tremendous amount of work. My team is part of the Krogan Lab at UCSF, and the work that I presented today was spearheaded by Chongyu and Ben. Specifically in the lab, we have close collaboration at UCSF with the Van Zastra Lab specifically, and I showed data that was generated by the Levels Lab at Cornell. So last slide, I just want to give a shout out. If you're an early career researcher, we are organizing a lot of events at the international HUPO level. And I thought this is a good opportunity to give a shout out to some of the competitions that we organize for early career researchers, such as the manuscript competition for postdocs and junior PIs and the thesis competition for PhD students. The application deadline is coming up May 13th, so you have still some time. And there's more information on the 2022 HUPO website. And I really would encourage you to apply for these competitions. And with that, I'm happy to take questions.
Thanks so much, Ruth. Yeah, so that's wonderful. It's quite a body of work. And yes, of course, US HUPO is very interacting with the international HUPO. So this is a perfect pitch you're doing here. I would definitely support that. It's great to, really also foster the early career people. Yeah, so I wanted to ask, so it's interesting to look at different tissue, different compartmentalizations. And so I was wondering actually 2 things. So have you ever done these experiments where you actually separated organelles beforehand? And then also, have you looked at similar pathways, but in different tissues? I wonder if in different organs, some of these networks are slightly different.
Yeah, I think these are two really good questions. So the first thing is that, we have, that's something that that we haven't done yet, or we have done it for a subset that we looked at the changes in. Now you can do the flip side, right? You can look at, for example, endosomal changes when we activate different receptors, and that's something that, especially now, seeing with the, just giving a snapshot of these multiple receptors that we looked at that are involved in pain, seeing that they have very different trafficking patterns to look at. Well, for the ones that actually go to the endosomes, how does the endosome changes? Do we see specific interaction that are recruited there? So we haven't done that yet, but I think this will be really interesting to do. So I haven't done that. Then you asked if different tissues or different cell types, and I think that's also something that I'm actually very eager to pursue in the future, because right now we have looked at only at HEC cells, and but what you can imagine, right, these receptors are expressed in neurons, which are very specialized cells, that these interactions might look different, and that's something that now that we have developed all these approaches, that we can start to apply this to more specialized cells. Like I'm starting to think about iPSC derived neurons and implementing it there.
Cool, great, thank you.
So stay tuned. Hopefully that will come at some point.
Yeah, next year we'll see that and hear that. Okay, then there was a question. Right?
Yeah, great talk, Ruth. And again, just to pitch again on the early career stuff, because that's actually how I met you, was virtually. For the first time was through that. So that's super cool. There's a couple questions. So the first is, are there any data analysis challenges when trying to calculate the location coefficients when you examine 3 different apex probes at the same time?
Mm-hmm. Yeah, that's a good question. So, I mean, we have spent quite a lot of time working on it, so it was not that trivial. But I think the main part is actually to set up this to characterize these spatial references very well, right? We have seen when we made the cell lines, that some of them showed like more cytosolic localization than we wanted to. And I think setting up the experiment right and characterizing the cell lines is actually more critical than the data analysis works actually quite well. Otherwise, you have to find ways to account for it. You can account for it, but we found that the most important part is actually setting up the experimental design well and characterizing these cell line well. The rest, the computational workflow that then so far what we have optimized works quite well. Then the other part is also to select the proteins that we start up with to predict the cell location and the coefficients that we did this pairwise comparison, but what we also did, we selected proteins that are consistently quantified across the spatial references, just to have, because we found that these are, if you have something that's consistently quantified, right, that allows to have a better prediction of the localization.
That's a great answer. Yeah. One more question, and if anyone actually has more, we probably have time for 2. This one is, did you validate the Apex technology for the nuclear receptors?
Yeah, good question. We haven't worked on nuclear receptors. I think that this could work similarly, right? That you would design if you want to see, right, we would label a lot of nuclear proteins that are maybe interacting, maybe spatial bystanders. So you would design also construct as a control that that has nuclear localization to do that. I think that should be possible, but I can only speculate, right? So we haven't done that. Yeah.
Great. Yeah, Ruth, I have another question. So, and I think I asked you this before in a different context, but it's just fascinating. Once you have set up the system, right, you can really look with quite high throughput at so many different conditions, drugs and time points. And as you demonstrated, what would you say is the effort you have to put in upfront, or that you saw you had to do to establish the system, and then how you really kind of harvest from there on?
Mm-hmm. So again, I think that's what I pointed out, generating cell lines that, stable cell lines that that are well characterized. Like you want to make sure that these constructs, these fusion constructs are functional, that they localize well. But then, I say we have now medium throughput way to do that. I think the biggest part is still the cell culture. The cell culture work is a lot of effort to do that. It's like I have great team members that can do that. They have set up a great framework to do that, but it's still manual, right? And we have this short time points. We take this we do the biotinylation just with adding hydrogen peroxide just for under a minute, and so that is something that I think is still something I would like to automate more, because I think that's right now the point where it's limited, right? Because that's still manual. So then I think the mass spec, the proteomics part is to do that over a large number of conditions. I think that using data independent acquisition, having consistent quantification was really key to help also with the data analysis, because you have less missing values, and you can then predict these trajectory over the time course much better. So I think that they're at different stages, different optimizations are required, but I think we invested now a lot in the proteomics part and the data analysis this part, and it's just the experimental upfront part, setting up a good cell systems to perform these experiments is crucial.
Thanks so much. All right.
Yeah, thanks again, Ruth. Thanks for coming and giving the talk. So our final speaker for today is Peggy Angel, who is also one of the co-organizers of US HUPO 2022. And yeah, thanks, Peggy.
Thank you so much, Ben. And Ruth, that was such a great talk. Thank you. Can everybody see my screen okay?
I think it's not.
Yes, we can.
There we go.
Yes, there we go. Okay.
Is that better?
Yes.
Okay, so Ben, you're doing a great job. You couldn't have picked a better host for this. So thank you again for allowing me to present my data. So I'm going to talk about differences in breast cancer post-translational collagen patterns between Black women and white women, particularly in South Carolina. And the last part of this work is really led by a College of Charleston graduate, Ashlyn Ivy. And so one of the great disparities in breast cancer is that although African-American women have slightly lower incidence rates, they have higher mortality rates. And in fact, it's around 36% higher mortality for Black women compared to any other race. And we know that if we look at how Black women are detected with cancer, we find that they're detected with later stages, higher grades, and more frequency of triple negative breast cancer. And so the mantra has been that aggressiveness is a primary difference in breast cancer of Black women. And so we know that there are challenges in socioeconomics and in medical access, but there's actually also factors that drive this increased aggressiveness, and that's what the field is currently exploring. We know that geographic origins can influence the breast tumor microenvironment. There's a number of geographically isolated groups and diasporic populations within the world that have their unique genetics. And when we compare African American origins versus European Americans, what we have found is that the generations of exposure have influenced particularly the immune system and development of an inflammatory response. And this results in different signatures in the tissue microenvironment and could be contributing to the disparity in cancer progression, which would affect, time of diagnosis, treatment, recurrence, and mortality. So what my lab is really focused on, and I hope you can see my mouse here, is looking at the contribution of collagen expression patterns within breast cancer that are higher in African-American women compared to European-American women. So when we look at breast cancer risk actually starts as an increase in breast stroma detected by breast density by mammogram. And so in these visuals, we can see this white pattern increasing. And what happens is that as the collagen stroma increases, we have a significantly increased risk of breast cancer, not only from production of the collagen stroma, but also it presents a challenge in detecting, because the tumor, as you can suppose here, is also going to be a white mass detected now within a white mass. And so if we look at molecular composition, on the top right here, we see a normal non-dense breast, mainly adipocytes, and then a high-risk dense breast tissue where the pink these are H&E stains, where the pink is actually collagen stroma. And so this is where you start getting the emergence of cancer-activated fibroblasts around the ductal glands and within this stroma. And so that's what we're interested in. And what we know from many years of studying is actually that we can develop these tissue-associated collagen signatures called TACs that qualitatively describe how these collagen fibers change during the progression of breast cancer, and they can actually be used to predict breast cancer. And so there's this very systematic way that is controlled by cancer-activated fibroblasts, particularly at the edge of the tumor, that results in metastatic breast cancer. And you can kind of see here by the lines drawing that we have this conversion from a tangential to the tumor border now to a parallel. And so this is frequently spoken of in breast cancer world as cancer-activated fibroblast highways. We also know that if we look, in fact, at many other aspects of collagen and breast cancer, so this is alignment shown in the previous image, but we can also look at post-translational modifications, particularly prolyl hydroxylases and collagen type. And so all of these features link to how breast cancer develops. They link to survival, they link to recurrence, but the challenge that we have now is that they really haven't been examined at a translational, post-translational level. They've only been examined primarily at the transcriptomics level. And so one of the important modifications that we're investigating in my laboratory really focuses on the collagen triple helical structure. So this is the center part of the collagen structure. There's a significant amount of control on how collagen organizes in the tissue and contributes to each organ's function. In addition, these sites of modification actually control how cells recognize and bind to the collagen as they move around, and they do cause signaling changes, particularly through the discoidin domain receptor and through integrin collagen binding. So what we have done is to access specifically the triple helical region of collagen is we're using unconventional enzymes, collagenase type 3. It has a secret little formula where I have helper enzymes that are added in very small percentages to help access these in formalin-fixed paraffin embedded tissue. And so we're looking at the center part of the collagen structure here, and we can detect not only the collagen structure, but also around 40 other ECM proteins from each formalin-fixed tissue section. And then this down here is the motif that we see by the collagenase type 3 expression pattern. So it is heavy for this triple helical region. So this is just a quick example of some of the collagen stromal proteins that we see from a single tissue of triple negative breast cancer. One of the things I would like to point out is for collagens, particularly the most abundant post-translational modification is hydroxylated proline. But if we look at other extracellular matrix proteins that pack around the collagen structures, those are going to be end-linked glycosylation and phosphorylation. So the PTMs are very critical in controlling how the cells move through the matrix, how the breast cancer reorganizes the extracellular matrix. We're also combining this with spatial localization using multi-imaging mass spectrometry. And so in this case, we take this same tissue section shown here, same slide that you would get from the pathologist. We prepare it with an enzyme. So now we're spraying it on the tissue, and we rapidly scan a laser across the tissue, and at each one of the pixels, these are in fact single pixels, we get a mass spectrum that reports hundreds to thousands of collagen peaks, and we can map each one of those peaks using software that allows us to select that peak and generate a heat map, and we can do pathological statistical testing on very specific regions that might be marked by the pathologist. So this is applicable to any type of tissue, but we're talking about breast cancer here. So this is the same tissue section with all the imaging mass spectrometry patterns. We take these and we can do hierarchical spatial clustering. And so each color here actually represents a collagen proteome, a collection of collagen peptides and other ECM peptides that are seen localized within the tissue. One of the study points I would like to make is what we are working to connect is that this collagen structure regulation is done by the spatial localization of a cell type. So it's not just the contribution of one cell, you have the contribution of other immune cells, inactivated fibroblasts and activated fibroblasts, as well as the tumor cells. And so when we go through and look at the domain mapping of very specific regions, this is actually shown here on liver tissue, we can find intense patterns of unmodified and particularly modified parts of the collagen domain within this tissue that clearly demark very specific cell composition. We can also follow it down because it's mass spectrometry. We can follow it down to the modified and unmodified structures, although there's a lot of work that goes into validating those. So this is an example of two breast lumpectomies, and what you can see here is that we have higher expression of this collagen 3A1 type peptide at the borders. This one happens to be triply hydroxylated. In this case, we have a collagen 1 peptide in green, which has one site of hydroxylation that is known. And then here we have another collagen 3 that is doubly hydroxylated. And I want to say one of the challenges in our research on these collagen peptides is that, you know, sometimes you get a peptide that has a single hydroxylation and you don't know if it's proline 1, 2, or 3. And so a lot of work goes into that. And there are spatial differences along which of these isobaric peptides are localized in tissue. So I'm just going to go quickly through it, but the point here is that we're doing imaging mass spectrometry. We then scrape the tissue and we're doing sequencing on the same exact tissue sections. And we do a lot of work with synthesized peptides, not only confirming the MS imaging, but also using those to understand cellular processes. This is an example from the Cancer Genome Atlas. And so here we are looking at self-identified breast lumpectomies from Black women compared to white women. And what we can see up here is a probability that each proline site is hydroxylated, annotated by doing max quant searches. And what we found is there's actually a number of proline hydroxylation that are varied. We can, try to make sure that we're not biasing things by comparing based on ancestry or race. And so if we did randomized tests here, the only comparison that showed significance was actually comparison by race. So, we're always trying to parse those differences. This is just an example of the data from LCMS-MS experiments where we're looking at these very specific sequence patterns. And I want to say here that, we're identifying these particular peptides. And again, in this case here, we have 3 prolines, and it could be that at a certain point when we're looking at bulk protein, there's a small group of cells that are actually having a different proline modification than is seen here. But we know that this one at least is confidently identified. We're always trying to get better at MS imaging. But in this case, the data also has transcriptomics. And we know if we just look at the transcriptomics, there's really not much difference between the collagen types. We know that prolyl 4 hydroxylase 3 is upregulated in breast cancer. There's a number of literature saying that prolyl hydroxylases are essential for breast cancer metastasis. But this doesn't tell us where the site of modification is occurring. And I think that that is what is important and what is the next thing in breast cancer. These are just some examples of image patterns. Again, here's a hydroxylated proline and non-hydroxylated proline, and you can see they don't completely overlap within the tissue. We can zoom in a little closer and we can see that this hydroxylated proline is actually at the tumor borders, right where the tumor is expanding and associating with the adipocytes. We've also found that this connects well with triple negative breast cancer. So this is a small group of samples from Black women and white women, 5 each. And here what we've done is we've gone through mapping first that there are spatial patterns of these particular hydroxylated prolines. This was done on a 7 Tesla FTICR for imaging patterns. And then second, that there are significant differences when we compare these between groups. So what we have actually done is we've gone back and in a lot of these sites, you can tell that the amino acid mutation contributes to disease. And so what we've done is gone and looked at these site modifications with the idea that if the amino acid mutation is causing a disease state change, then maybe it's more likely that a post-translational difference could also cause a change. And so for the ones that I showed on the previous slide, actually 3 of those actually map to sites where there are also contain an amino acid modification. And I know amino acid modifications represent much larger structural variation than hydroxylated proline, but this would go well. This goes well with our cell studies where we're looking at synthesized peptides and their effects on cells. I'm not going to talk about that today. So now I want to talk a little bit about structure regulation in the South Carolina Black women and white women. Again, this is done by Ashlyn Ivy. So in the United States, we have around a 36% increase in mortality, but when we look in South Carolina, we actually have a much higher rate of mortality in Black women in South Carolina. So we can look along the borders of the coast, and we actually have an NCI designated cancer center here that decreases mortality compared to, the mainland non-coastal regions of South Carolina. However, the mortality is still significantly increased in these women. And so we want to know, it's not just access to an NCI designated cancer center that has embedded well with the community. It's, really like, is there something different that we can maybe leverage to identify risk populations? In South Carolina, we have a unique population of Gullah Geechee people. So these people were brought over from the West African coast during slavery. And so these groups, which are shown here in red, have formed their own nation among the coastal regions, and they have much higher levels of African ancestry, particularly in common with the West African populations. And so there's a really interesting paper done by the researchers here at MUSC. And so they have defined genetically these groups of people that, as far as their West African compositions. And remember, West African composition is one of the higher risk for more aggressive breast cancers. So this is just an example of what I had just reported. And so there's a numerous amount of studies looking at East Africa versus West Africa. And we know that particularly from these Windward and Gold Coast regions, from which the South Carolina Gullah Geechee have settled, we know that those regions in particular have much higher rates of triple negative breast cancer. So they're associated with both subtypes, aggressiveness of breast cancer, and a differential tumor response that contributes overall to higher mortalities. So in this case, we were able to, working with Marvella Ford, who has integrated with the Gullah Geechee community, we looked at newly diagnosed breast cancer. And in this case, we're using both parents had to be born in the Sea Islander geographical region. And the median age of the patients themselves was 59. And so we estimate that the parents who were born in the region have probably been at least in the region since at least I would say the 1940s or 1950s, but frequently longer. So in this case, we're looking at tissue microarrays. And so this is a unique modality to mass spectrometry imaging. So we have cores that are collected, directed by pathologists from the tissue, and they're arranged in arrays. And so we can do the deglycosylation, which was published on this group in 2019. And then we again spray it with collagenase, and we look at these peak intensities by accurate mass to try to understand the differences in the population. So what we found was that the tumors show low numbers of peptide variation among the South Carolina Black women and white women. This is something that's actually been repeated in a number of different experiments at the Hollings Cancer Center. And so the Gullah Geechee actually have a very healthy lifestyle. They live in communities, and they actually eat probably a lot better than the mainlanders. But so this isn't really surprising, but what we did find is that we had very key specific elevation of certain peptides. And here I'm just saying what the protein level does. I'm not saying what these particular identified prolyl hydroxylase modified collagens do. But in specifically, I want to point out collagen 3A1, which is a target of recent cancer research. So this is associated with cancer stemness in African Americans. So we also looked at tumors independent of race, and what we saw that there was a difference in reoccurrence and occurrence in South Carolina women. So we could actually look at the collagen structure variation, and we can almost predict who had reoccurrent or who did not have reoccurrent breast cancer. And again, these are led by peptides that are modified by prolyl hydroxylase. In normal adjacent tissue, we actually saw a larger variation than compared to the tumor tissue. One of the things that we are finding using other glycosylation studies is that Black women have a more pro-inflammatory normal tissue. We also know that we had changes in collagen 3A1 again, so seen here where it was decreased in Black women, and we know that collagen 3 generally, again, I'm not talking about specific components of the structure, actually contributes to proliferative breast cancer. So in the TMA, we also had breast lymph. So this is a little unusual. And I was like, okay, let's go ahead and take a look. And so we know in recent literature, there's pretty big differences that are found in lymph nodes that are invaded versus non-invaded, and in triple negative breast cancer. And one of the key signatures that changes is the extracellular matrix. And so looking at these, we did have a large variation here. And as far as I know, this is the first time someone has looked at the collagen proteome in lymph. And so in normal lymph, and I want to say too, that there's some differences in normal lymph clinically than in between Black women and white women. So it's just great in this TMA that we were able to compare to normal lymph, where we saw some difference between just both normal and then also comparing, in this case, we had differences in white women normal lymph versus metastatic lymph, which is marked here. So there were several differences in only metastatic lymph. And so those are shown here. And so those particularly related to collagen 1A1, although there was a number of peptides. And the ones that I'm showing here are elevated in Black women. So it appears that the collagen regulation, not only of the tumors, so we have the tumors, we have the normal adjacent tissue, and then we have the metastatic pathways that these tumor cells take. So these all appear to have some sort of modification related to them. And what we think is happening is there's collagen post-translational regulation that provides these tipping points between progressing from a dormant to a metastatic case. And, that's something that we don't know. We're always basically staining for the intact collagen protein or looking at it by transcriptomics. And then last, one of the exciting projects that we've recently embarked on with Hari Krishnan Nakshatri is looking at the Duffy antigen receptor for chemokines, also known as the atypical chemokine receptor. So this is a receptor for Plasmodium vivax, which gives resistance to malaria, and it's found mainly on the West African coast. So it confers a protective immune feature to these to the people of the West African coast. And so what we have found too, looking at these characterized by heterozygous or homozygous Duffy, is that there are significant changes in breast tumor. So T here is tumor, and then N is normal, where we had a lot more variation in normal for both mainly for the heterozygous, but you can see that there was also less samples. But in the tumor, we had a pretty large number of samples, and there were significant differences there. And so we know from previous publications that Duffy does increase the macrophage and T cell receptor. There's an association with increased incidence of triple negative breast cancer metastasis. And so what we're hoping to do with these studies to kind of conclude here is to use these points not only to find out new features of aggressive breast cancers, but if we find these features in particular at-risk populations, maybe they can be used for other people within the United States or the world that could have high risk. Maybe there's a marker for more aggressive breast cancers that could change the way treatment decision is made. So another population that we're looking at is actually the Appalachian population within Kentucky, and they also have very high risk of breast cancer. And so just to summarize, it's possible that these post-translational modifications could contribute to aggressiveness of breast cancer in Black women. And I think that the spatial distribution is key to this. You know, is it occurring at the tumor borders? The Sea Islander study shows kind of a new region for me. Maybe I'm being silly, but I think that looking not only in the breast tissue, but also in the lymph is going to tell us a lot more about what propels or allows metastasis, right? So there could be some really key proteomic differences there that need to be studied. And I really advocate that we do more population proteomics of isolated or diasporic populations. And I think this will help us understand a lot of, cancer controls, going through diagnosis, prognosis, and helping with treatment decisions. So I specifically want to thank the patients and the donors. Without them, it's a scary time to have cancer, and, to have someone ask you to donate your tissue can be kind of weird. And so I really appreciate their contributions, as well as those of my lab and close collaborators. And so I will take any questions.
Maybe, yeah, thank you, Peggy, so much. This is just fascinating. It fascinates me every time again when I see this, and so, yeah, so how long does it take actually you showed some of these slices and images. How long does it take to going from when you spray to when you see, and the mass spec part?
Yeah, so it really depends on what analyte you're looking for. If you're looking at glycan patterns, it is very fast. The digestion is 2 hours. And so then we spray a multi-matrix on it and we can scan it. Now the scanning is very fast now. There's probably around 17 to 20 or more pixels per second, depend on how you have tuned the instrument. So it's very fast. So for a whole tissue section, it's around 40 minutes. And it used to be like 3 to 5 hours. So things have sped up considerably. There's a lot more control over the lasers. And then because we're using sprayers, we've kind of almost eliminated technical reproducibility. And especially for a TMA where all the cores are on one slide, right?
Yeah. Great, thank you. And that's really impressive. And Ben, I think there was actually a question in the chat.
Yeah, so we've got 3 questions so far. And yeah,
Yes.
Peggy, thank you again for giving this talk. I missed this talk at US HUPO, so I'm really glad to get to hear it. It's
And
really
it's always
awesome.
nice to see you.
Yeah, really, really good stuff. So our first question is, do you know if the collagen PTMs are a cause or effect of the disease progression metastasis?
That's what we're trying to find out. So, what we're doing is taking a spin on how they look at collagenase digest, but we use through regenerative medicine approaches where they fractionate it. We're more specific because we have one part of the collagen structure we can study, right? So we're taking that and putting it into, cell culture media and into the ECM that's used for cell culture. And we know that there's definitely proliferative changes based on where the site is modified. And so I'm hoping that maybe by ASMS, which is in about 2 weeks, 3 weeks, I guess, maybe I can show some of that data. But the challenge is collagen is actually a triple helical structure, right? And so we're spiking the single peptides in almost like a drug. So there's always need for more tools and to get better exact, more exact structural microenvironments to study.
And just, I'm going to ask these two questions as well, but real fast from me. I mean, because you have this specific population, did you spend, or was there concern with the databases, like the genomes, like the sequences themselves?
So there's no data on the sequences from these women.
Okay.
There, as far as like the genetic sequences. So what they've done is they've done a 100-point SNP to actually pinpoint their ancestry to Senegal, right? So it's not just Africa, it's not West Africa, it's actually to a very specific region in Africa. And that was done by Paula Ramos at MUSC, and that's a really interesting paper to read. Cool. So we don't have the genetics on that particular group.
No, that's still great. I mean, it's a building story, right? Okay, so 2 more questions. So again, great talk. For collagen PTMs, is there a way to enrich hydroxyproline modifications or enrich collagen? And can you provide some insight about the structural roles for like hydroxylysine versus hydroxyproline in collagen?
Yeah, so hydroxylysine is a cross-linking enzyme that that activity occurs in the extracellular matrix. And so the cell is extruding these little bricks of polypeptides that then get assembled in the extracellular matrix, and LOX is one of the enzymes that connects them with other ECM proteins and with each other. Prolyl hydroxylase is actually done in the early endoplasmic reticulum, and so that's done as these little bricks of polypeptides are prepared. And so I guess it would probably relate to metabolomics and proline metabolomics and in ER stress, like everything in the PTMs. And those particular sites actually influence how the cells recognize the ECM, and they control their signaling patterns and the molecular patterns that they're putting out to nearby cells.
Wow, that's really cool. I think a little bit about hydroxyproline, but never knew it did that much. There's actually 2 more. So have you observed increases in collagen 1 peptide signal in the tumors? And are there plans to combine imaging mass spec with SHG for collagen architecture?
Yes, and actually we've done that. So SHG is second harmonic generation, and it's a microscopy method that uses photons to measure how collagen fibers are arranged and also their size and length. And so we've actually created workflows that allow us to do H&E staining first. We send them off to our collaborator in Nebraska, Heather Jensen Smith. And so she measures by SHG the collagen fibers and sends it back, and we remove the cover slip and we do imaging on the same sample. So that kind of combines the arrangement with the proteomics.
And they actually clarified, because I misread, I should read better, Col1 propeptide signal in the tumors.
I think that might be harder because And maybe I'm mistaken, but the propeptide would be part of the N terminal of the collagen, or C terminal. There's a signaling collagen peptide, or it's not signaling, I shouldn't say. There's a biomarker peptide that's on the C terminal that's called ProC3. So that's why I'm saying that. But our enzyme actually has a very heavy and specific activity on the central portion of the collagen structure. And so you can see that in some of our some of our posters where we show the sequence coverage of this. And so it's not hitting those N terminal, C terminal regions. So I don't know if we would see that. I think combining enzymes would probably be the way to go.
Cool. And I think we're going to do last question. So did you try to correlate the presence of MMPs with the increased abundance of MMP-derived collagen peptides in the metastatic potential in the imaged breast tissue areas?
No. So what we're doing is we're actually purposefully digesting the tissue by collagenase. So it would be hard to say this peptide is a combination of an MMP and a collagenase. So that's kind of a whole. But one of the things that we have done is we've developed MMPs that target human they're human MMPs that target human collagen. And so we can actually use those to digest the structure. The other potentially confounding technical thing is these are on formalin fixed paraffin embedded tissue as associated with clinical tissues. And so we do work to remove the cross-linking structure, and there's plenty of work that's done that says you can almost nearly completely remove it. But I always keep that in mind that, there could be some remaining, but we haven't purposefully done tried to look for an MMP produced peptide. That's a good question.
Thank you again, Peggy. Birgit? Birgit, you're muted.
All right. Yes, I was muted. This is wonderful. I mean, this was such an inspiring afternoon, and thank you, Ben, for managing this, and to all our speakers. If you like to turn your videos on, I mean, I really would love to thank you. And Susan started us off strong, and everybody brought so much enthusiasm. And I just loved that we saw like really very cutting edge technologies, but then applying it to really super interesting biology. And I think that's really the strength of proteomics, that it can combine these two aspects that seem different, but they really merge together. And that's maybe why I like to be so much here. And I will write a little synopsis of this, day to day, and then we'll post the video and other people can also see it. I don't know what you think, but I thought this was just as fun as our real life conference. And thank you, everybody on the screens at home or at work. And Brianna, if you want to show us, we have our next conference in person in Chicago, and I would love for all of you to join and come. We have good hotel rates and just fun to hang out with the US HUPO scientist. Right, Ben? You have the last word, Ben. You can say.
Oh, I have the last word. Well, I mean, I want to thank everyone. I mean, again, thank you, US HUPO. Thank you for doing this in Charleston. Peggy was one of the co-organizers. And thank you, for our best of. I think this was important for people that couldn't come. And I appreciate you making time again to do it. Yes. So thank you all. Really, this means a lot.
Thank you.
Thank you.
Thank you.